I'd like to welcome everyone to the annual Penn Institute on Aging and Center for Neurodegenerative Disease Research Retreat. It's a special and exciting day today for several reasons. This is the first integrated research symposium for the IOA and CNDR, which previously held separate complementary research retreats each year. The theme of today's retreat is about coming together to integrate the discoveries in basic science and translational research in age-associated neurodegenerative disease, with the overall goal of improving human health. The theme of coming together is even more important now and timely as we're emerging from the COVID-19 pandemic. And for many of us, this is the first time that we've been in person together. Several of you I haven't seen in months other than a TV screen, so it feels great to see everyone. I'd like to thank Dr. John Trojanowski, who's the CNDR co-director and former IOA director, and Dr. Virginia Lee, who directs the Penn CNDR, who gave me the opportunity to organize the event today. I'd also like to recognize John's exceptional leadership in the Penn IOA for nearly 2 decades, which has led to a wonderfully collaborative and rich academic environment here at Penn that truly sets us apart. The theme of this retreat, to integrate scientific backgrounds that are diverse to improve human health, aligns well with the mission and vision that John has for Penn IOA and the collaborative atmosphere by John and Virginia have created here at Penn. So we're thrilled and welcome to thank our keynote speakers, Dr. David Bennett, who directs the Rush ADC and has pioneered research into preclinical AD and mixed pathology and population-based cohorts, and has been a pioneer of open science as well, and Dr. Leonard Petrocelli, who's the chair of neuroscience at Mayo Clinic and discovered key pathological features of C9orf72, expansion form of TDP43 proteinopathy, and leads a translational program to develop novel therapeutics and biomarkers for FTD and ALS. Finally, I'd like to thank Dr. Kathy Jadrewski and her team at IOA, including Ebony Fenderson and Nicole Calvecchia, who has been instrumental in organizing all the activities today and the logistics. So we have an exciting program today, and I want to get started by introducing our first speaker, who is Secretary Robert Torres, who kindly joins us today. Robert Torres was appointed by Governor Tom Wolf to serve as the Secretary of Aging in 2019, has been serving as the active Secretary of the Commonwealth since October 2017. He was confirmed in 2019, and we welcome you here this morning and look forward to hearing from you. Thank you so much.
Good morning, everyone. It's a pleasure to be here. I, too, am enjoying getting back to some semblance of normalcy. Before I begin my remarks, I just want to acknowledge the presence of Mr. Nadja Orr, who is President and CEO of the Philadelphia Corporation for Aging. Nadja took over the Corporation for Aging right at the beginning of the pandemic, and there's a lot of changes going on there, and he's looking for partnership and collaboration opportunities. That's why he's joined us. And Tom Sneddon, who some of you know, is the director of our PACE Pharmaceutical Benefits Program, who's here with me as well. So I'm here to give you a quick update on our state plan on aging. Before doing so, again, I also want to thank Kathy for extending the kind invitation to be with you. This state plan on aging was something that we're required to do every 4 years. And basically, the purpose of the state plan is to establish a mission, vision, and core values of the department, which we worked on for over a year. We actually started in the summer of 2019, knowing that we would have to produce a new state plan on aging, engage stakeholders, and I'm really proud of the work that we did, and especially because we were in COVID, we were able to engage virtually with over 500 stakeholders across Pennsylvania. I doubt seriously that would have happened if I was traveling around the state, talking in front of audiences. So we really got a lot of good input. Part of what we had to do was review our structure, our area agencies on aging, or AAAs as we refer to them, the councils, and different programs that we administer at the department through our area agencies on aging. We had to take a look at our demographics in terms of older adults, individuals over 60, and then really look at emerging trends and figure out, okay, what are the trends that we have to be sensitive to, and what are we proposing in our plan. Then we had to put together goals, objectives, strategies with measurable outcomes over the next 4 years. And actually, this is a state plan, so there's a kind of consumer-friendly version of it. The actual plan was over 100 pages that went to the federal government, but this is online, but if anybody's interested, please feel free to contact us and we'd be happy to send you our hard copy. And again, the plan provides a blueprint for the next 4 years, establishes outcomes and that we are planning to work on and achieve, and looking at best practices, trying to leverage the work that we do to help build capacity. Right now, Pennsylvania's over 60 population is over 25% of our total population in Pennsylvania. Over the next 9 years, it's projected to grow another 9 million. So that's going to be 30% of the total population of Pennsylvania. So again, with that kind of growth that we've seen over the past decade and that we'll see throughout this decade, you can anticipate there's going to be a large demand for services. The Administration for Community Living, or ACL, required us to look at 4 primary focus areas. One was looking at the programs that are funded and supported by the Older Americans Act. That's our caregiver support program, nutrition, employment for older adults, and strengthening our network capacity to provide services for seniors. They also wanted us to concentrate on programs that help with discretionary funding. So most of our funding in Pennsylvania, 81% of my funding is lottery money. So when you hear, the lottery supports seniors, it really does. 81% of my budget is lottery funds. The rest is primarily federal dollars. But, I've always encouraged not just my department, but the area agencies on aging to really look at diversifying funding in case at any point, that funding gets compromised in any way. And the discretionary funding, again, looks at, issues like Alzheimer's, health and wellness, age-friendly communities, and no wrong door programs to support older adults. A lot of emphasis goes to person-centered planning, making sure that we're really paying attention to the needs of older adults and connecting them to appropriate resources. And then a big part, and probably one of the most important things that we do, is elder justice, protecting older adults from elder abuse. That could be physical abuse, neglect, abandonment, or exploitation, financial exploitation, which we've done a ton of work on. And we provide ombudsman services, which were very active during COVID, because these are individuals who are advocating for the rights of long-term care residents. So we have 5 state goals. The first one is strengthening our network's capacity, promoting innovation and best practices, and really looking to build efficiency. Again, I told you how much the population is projected to increase, so we're really thinking through how do we expand our capacity to meet the demand that's coming. And that's here, really. We want to improve services for older adults and the ability to advocate by using evidence-based information, really committing to data integrity, and being accountable for results. So we've been doing a lot of work in terms of improving the accuracy, integrity of our data, better analytics. We're even over 20 area agencies on aging have joined health information exchanges, so they're able to see health records on their consumers, and the healthcare professionals are able to see care plan data, which is one of my personal priorities. I really want to see better alignment and coordination of health and social services, so they're working together to best support the older adults. Want to establish and enhance our efforts to support healthy living and active engagement, really give older adults a sense of purpose and a sense of community, so that they can age well and, as independently and in their homes and community as long as possible. Emphasizing a citizens first culture, which means doing effective outreach, making sure that we're reaching populations that are underserved and perhaps we have not been as effective at reaching in the past. Embracing diversity and really honoring individual choice. And then the last one has to do with elder justice, which again is advocating for the rights of older adults, making sure they're being safe. We're doing a lot of work in terms of predictive analytics, looking at the data that we have and really trying to anticipate and be more proactive in terms of helping older adults not be victims of elder abuse, especially neglect, self-neglect, or exploitation. So the themes basically revolve around access, improving our outreach to older adults. We did a lot of work related to social isolation, and I'll talk about that in a second. Increasing our capacity to meet the anticipated demand that's going to be here over the next 9 years. offering better protection. Again, ton of work that we did and published on financial exploitation. All this information is on our website. And then a lot of work around diversity, equity, and inclusion. So in terms of improving access during COVID, we utilized technology as much as possible. This past Tuesday, I was actually at Bucks County at a senior center promoting booster shots and the flu vaccine. And while I was there was a gentleman that was going around helping seniors, basically giving them iPads, teaching them how to use it. And then I was able to sit with 3 seniors and hear how the iPads have changed, their life for the better in terms of keeping them connected with their families and friends. One lady said, well, there's one problem with the iPad, now I can't sleep. And I said, why? I said, you're addicted. And she said, yeah, I have to get off the iPad now. So they're really using it, and we're promoting it as much as possible. We have university partnerships that I'll talk about in a little bit, where college students, both undergrad and grad, are working with assigned seniors. And this semester, we actually have a program where if the senior is interested, we will lend them iPads to work with the students. And at the end of the semester, those are loaned, but then we have another grant program where if the senior is interested in maintaining technology, we can give it to them. We're working on broadband in the rural parts of Pennsylvania. We're working with electric cooperatives and high school students, again, to help train seniors and really try to bring the resources in the rural areas. Doing a lot of work with telehealth, telemedicine, virtual senior centers. So all these things have really been promoted and moved forward as a result of COVID and the fact that so many were not connected and isolated. Health information exchange, HIE, I talked a little bit about that. AAAs now can have access to their consumers' health information. They know future appointments. They get daily messages on whether any other consumers have presented at the emergency room, if they've been admitted and discharged. So that helps the area agencies on aging, because some of them are sending people out to deliver food, to do home health, and now they know that, okay, this person's going to be out for a week, they're admitted at the hospital, they can adjust their resources and not have people driving out and not have the consumer there. And again, my big thing is I really, really want to see the integration of health and social services, and that's why we've been working pretty hard over the last 2 years to get there. In terms of diversity, equity, and inclusion, the Older Americans Act, so the state plan that we put together requires that we give particular emphasis to communities of greatest economic and greatest social need. So the greatest economic need is those individuals over 60 who are living at poverty or below. Greatest social need are communities that are isolated for different reasons. So it could be underrepresented races and ethnicities, people isolated because of their rural geographic area, people that are limited English proficient. So maybe that causes some isolations, or people living with a disability. And we have been doing a lot of work with LGBTQ elders. And early this year in January, I developed a policy directive for Pennsylvania declaring the LGBT community as a community of social need because of the isolation and the challenges that they face. To work with to support these efforts for underrepresented races and ethnicities, the governor has different affinity commissions, so the African American, the Asian Pacific Islander, Latino, LGBT. So we work with all of them. We've been trying to wrap and really coordinate our efforts with groups that can support us. We go into communities with, work with community leaders. We have Language Line. We're translating more in terms of materials that we have. PACE Call Center does a great job with limited English proficiency. And in terms, again, of the LGBT community, we're starting to collect more data. So again, that'll help us down the road as well. Social isolation. So again, once the pandemic hit, all senior centers in Pennsylvania closed. So that's over 500 senior centers that closed down, and these seniors stayed home, many of them by themselves. So social isolation became a top priority. We were already working on social isolation issues as a priority. COVID just accelerated the work that we were doing. Our Pennsylvania Council on Aging actually developed an online guide, and that council is primarily older adults, so they really wanted to to do something to support older adults. And again, if you go to our website, you'll see the solo guide. And it was just a tool to help seniors, stay healthy mentally, spiritually, emotionally, and so on. A lot of the AAAs did great work in terms of being creative to support older adults. We started an intergenerational university program with Slippery Rock that has gotten a lot of attention and has since expanded. Widener University and Westchester University in this region are two of our newest ones, and I'll be at Westchester Tuesday doing a press conference with students and their assigned older adults talking about the program and the benefits that it has brought over the past 2 years. Doing a lot of data collection, again, trying to assess risk very early and try to do more predictive analytics in terms of things that we could do proactively instead of being reactive. And then ADRC, and I apologize for these acronyms, this is our Aging and Disability Resource Center. We have partners, organizations that provide services to seniors, and this is kind of a central place for an information and referral. But they got a grant from Temple, they got a grant that we're using to support Temple University's called Tech Owl. That's where we're getting the equipment to work with the seniors. So that particular grant is helping us to fund the university students that are working with their assigned seniors. In terms of protection for vulnerable older adults, our top priority is getting what's called the Older Adult Protection Services Act updated. It's been lingering for years, and it's something that I want to see done before this administration comes to an end, because it hasn't been updated in more than 10 years, which is a shame, because a lot of things have changed and we need to get that particular legislation passed. Financial exploitation, we did a study, we issued it last September, and we continue to work. So we put out recommendations, we have small work groups, and in January you'll probably see another report coming out with the progress we've made in implementing those recommendations. We're examining all our processes to, again, be more, see where we can be more efficient and more productive in getting ahead of some of the problems that we see. We developed and implemented new mandatory reporter training. Again, that's all on our website. We've improved our call center operations so that people get live bodies, not just an answering service. And we have a new public-facing, what's called report of need. So when people call in, we fill out a report of need for protective services. And our ombudsman during COVID did a lot of really good work trying to help residents of long-term care facilities stay connected. They even use technology. They use a pad bot. So it was basically an iPad on a little robot that they could navigate in a nursing home with their phone and be able to talk face-to-face with their residents. In terms of building capacity, again, university partnerships are important. Like this one, I borrowed these slides from my special assistant, but Penn should be up there because you're an important partner for us as well. But I mentioned the ones that we did during COVID. We're having conversation with Penn State University College of Nursing. They're doing a lot of work with geriatrics and looking to really see how they could apply with us. Working with Temple Institute on Protective Services and, again, Tech Owl. Working with federally qualified health centers. When I got to the department, there wasn't a lot of interaction between the department and federally qualified health centers. So trying to see where we can work together to support older adults that are, getting services there. And then we're working a lot, and I think, I've worked for the last 3 administrations. This administration has really done a good job of breaking down the silos. So I work, regularly with Department of Health, Human Service, Drug and Alcohol, and we do a pretty good job of coordinating our efforts and supporting each other. Things like the Opioid Command Center. Tom, with his PACE program, is helping Department of Agriculture with like Senior Food Box program, because we have, we're taking advantage of the resources that we have to help each other. Final thoughts. So our mission is to promote independence, purpose, and well-being in the lives of older adults through advocacy, service, and protection. And our vision of Pennsylvania, where older adults are embraced and empowered to live and age with dignity and respect. I do want to mention we've done a lot of work with Alzheimer's. I don't know if some of you saw the article from Spotlight that said that was very critical of Pennsylvania's level of preparedness in terms of dealing with Alzheimer's and dementia, but we've done a lot of work. I was a little disappointed. I get the article from a standpoint of advocacy, but I think over the past 2 years, we've done a lot of work. We've worked with the Jewish Healthcare Foundation in Pittsburgh and have over 7,000 people that have been trained as dementia friends. We have several hundred people in 51 counties that are dementia champions. Department of Health, you'll be hearing from Dr. Woke later this morning. He got a $3.5 million grant to work on Alzheimer's. And, again, on the theme of diversity, what are the implications for African Americans? So a lot of good work going on, and I wish you an excellent day today, and thank you again for having me at this retreat. And again, feel free to contact my special assistant if you'd like hard copies, or we can help in any way. Thank you.
Thank you very much, Robert, for coming and joining us this morning. That was terrific and aligns well with the mission of Penn and IOA, Penn Memory Center, and our groups of reaching out for resources for aging and related cognitive impairment. A few housekeeping items as we move forward with the agenda. Our first series of talks in mechanistic work and basic science and neurodegenerative disease research. We will have a Q&A section for those who are virtual that we'll be monitoring if you have questions, and also in the audience for questions we can, I'll be able to repeat it through the microphone for you so that it's recorded. So now it's my pleasure to announce our keynote speaker, Dr. Leonard Petruccelli. He's a consultant and enterprise chair of the Department of Neurosciences at the Mayo Clinic. He holds the academic rank of Professor of Neuroscience in the Mayo Clinic College of Medicine and Science, and is recognized with the highest distinction of the Ralph B. and Ruth K. Aperson Professorship at Mayo Clinic. His research team is focused on researching cellular mechanisms of ALS, FTD, and uses a diverse set of expertise in drug discovery, cell biology, and iPSC modeling. And the overall goal is developing new biomarkers and treatments for these disorders, including C9orf72. He's a principal investigator of several grants funded by the NIH, including a program project on FTD and ALS, as well as a Tau Center Without Walls. So Dr. Petruccelli, it's wonderful to have you here this morning, and look forward to your talk.
Thank you very much, David, and for the very kind invitation to allow me to present at your annual retreat. And it was just, fantastic to see so many familiar faces yesterday that I haven't seen in quite some time. So while this is a virtual presentation, I really encourage, the audience as well as the guests who are participating online to interrupt me throughout my presentation if you have any questions, trying to keep this as informal, as possible. And so I'll simply have my disclosures as shown here. And I really enjoy starting off my presentation somewhat with a timeline, because I think it really highlights the accomplishments that we've made so far with major genetic discoveries starting in 2006 with the identification of the progranulin gene being associated with FTD on chromosome 17, but also our efforts, I think, through collaborative efforts, not only with academia, but also in industry, where we've made enormous strides in translating our basic science programs and neuropathological observations into the clinic. And so, I think C9 perhaps represents somewhat of a textbook example of how through that, discovery, and we actually now have an active clinical trial led by Biogen on this. So, it did start, with the identification of the progranulin gene, as I mentioned, and of course the work by Virginia and her colleagues at UPenn that identified TDP-43 as, a major pathological feature for these diseases, and it changed the field in an extremely profound way, not only for individuals with sporadic ALS and FTD, but also really, although it was a pathological observation, it really started to put the framework, mechanistic framework for how TDP-43 may be causing the disease. Subsequently, other RNA binding proteins such as FUS was identified, and in 2011, a hexamer repeat expansion called C9orf72, was identified as a major mutation associated with familial ALS and FTD. And nearly 40 to 50% of those cases can be explained by this mutation. And then subsequently, which will be a portion of my talk today, is the significance in the discovery of these abnormal proteins that accumulate as a result of this repeat expansion. So Rosa Rademacher at Mayo Clinic, as well as Brian Traynor at NIH, simultaneously published back-to-back papers in Neuron, identifying how this very large hexanucleotide repeat expansion consisting of GGGCC was identified. They're very long and large repeats, as you can see by the Southern blot analysis, and consistent with what you also find with other repeat expansions are these very distinct RNA foci that accumulate throughout the brain in the nucleus. And so, that in part, led to an initial framework for, how, this repeat expansion may be occurring, as well as a reduction in C9 message, since the mutant allele containing it, is compromised and leading to a haploinsufficiency of the transcript. We became, interested in this following up a very provocative idea put forth by Laura Ranum at the University of Florida that, sometimes these repeated expansions could adopt a secondary structure very similar to like a stem loop structure, and that in itself could potentially initiate protein translation in a very unconventional way, independent of your typical Kozak AUG start site. So we were very intrigued with this idea and followed up on it from an antibody-based approach and simply asked, if in fact this 6 nucleotide sequence could generate peptides, what would it be? And let's generate antibodies against them. And so as you can see here in the sense direction, you would have these repeating amino acids, amino acids of glycine alanine, glycine proline, glycine arginine. And if in fact that this mechanism also occurred in the antisense direction, 2 additional dipeptide repeats of proline arginine and proline alanine would in fact be present. So as you can see here, what we fundamentally observed is that these abnormal protein products, all 5 of them can in fact be detected in brains that harbor the C9 mutation. You can see here antibodies against the PA and PR peptides can be detected throughout the hippocampus and cerebellum, as well as other brain regions. But these are by far the most least abundant. The sense dipeptide repeats, in particular GA and GP, are by far the most abundant, followed by GR. So we use these, antibodies to really, tease out, excuse me, additional studies in postmortem brain, but it also added another layer of complexity associated with C9. And that is, what in fact are these, very unique dipeptide products doing? Are they simply innocent bystanders, or are they, participating in the pathogenesis of the disease? And so we and others, utilize cell culture systems, fly models, as well as animal models to really understand what these protein products are doing. But once again, C9 is perhaps a very unfortunate neurodegenerative disease from a pathological standpoint because of the fact that it's very heterogeneous. In addition to C9 pathology of those RNA foci and those dipeptide repeats, fundamentally these patients unfortunately also have TDP43, where normally it's within the nucleus, and for reasons which we don't understand is mislocalized and forms these very distinct and profound cytoplasmic inclusions. I would say, and the reason why I'm emphasizing this here is because, my lab as well as other labs really focused initially on, the significance of these cytoplasmic inclusions. But I think now we're really paying attention to what is the pathological consequence of having the nucleus devoid of TDP-43 expression, and whether or not that this might provide opportunities to understand novel mechanisms associated with the pathogenesis of the disease, but also potential biomarkers, which is what we're also very interested in as well. So, based on that, what is, that link between C9 and TDP-43? And so I'm just capturing some of the studies that we've done. I think perhaps the most exciting studies were the ones that we've conducted in vivo, in which we were able to, generate a repeat expansion that was initially of 66 repeats, but the data shown here was one of 149 repeats. And so we knew that if we were to overexpress this based on our cell culture systems, that we would anticipate that we would, see RNA foci in vivo. And so what we did was we took advantage of an AAV-based approach. We injected animals at P0, so before they're 24 hours of age. And then we collected and harvested the animals and interrogated them for pathology at 3, 6, and 12 months. And as you can see here, based on this FISH analysis for the sense as well as the antisense strand, you can see that very distinct transcripts, in fact, do accumulate in the nucleus of these animals really throughout the brain, and that's the advantage of the AAV. And so here's just the analysis that we conducted in the motor cortex as well as the hippocampus, where we actually do observe an age-dependent increase in the number of RNA foci in the brain regions highlighted here. So is it possible that these animals, which are expressing this hexanucleotide repeat, in fact, able to have this unconventional form of protein translation in vivo? So using the antibodies that we made and that we used for post-mortem human analysis, you can see at 3 and 6 and 12 months, very distinct staining for each of the individual dipeptide repeats shown here. They do accumulate in an age-dependent manner, as shown here for poly-GA and poly-GP, and this is the analysis that's shown here. What's perhaps most important, at least for me, is the fact that the amount of the proteins, recognizing that this is an overexpression system, but what you do find is that the abundance of these respective dipeptide repeats features what you actually find in the human brain. And what I mean by that is that GA and GP are by far the most abundant dipeptide repeats in our animal model, and that's exactly what you see in human brain, followed by poly-GR, and once again, PA and PR being the least abundant. They could be abundant because of the fact that the antisense transcript is less abundant, so therefore there's less templating. The unconventional form of protein translation may be less efficient, or some of them may be simply just incredibly toxic, and work by us and others have shown how PR in particular is incredibly toxic to the cell. The result that was perhaps the most gratifying was, whether or not TDP-43 pathology would exist. And so what we've shown here using a phosphate TDP-43 antibody is that we start to see as early as 3 months some distinct puncta inside the cells, but very in an age-dependent manner, you can see that these phosphate TDP-43 inclusions become quite prominent at 6 months and 12 months. And so this is, for us, was incredibly exciting because of the fact that, we were able to detect, the endogenous, phosphate TDP-43 cytoplasmic inclusions in vivo versus, overexpression, models that, have been generated, including work from our own lab. So we're still, aging these animals, and we're still, continuing a lot of work, following up on this initial observation. And so, as the field was emerging, we became really excited about the potential use of antisense oligonucleotides. And and we and others, the work from Don Cleveland and Clotilde, really participated in testing these ASOs in their back transgenic animals as well as their iPSC culture systems. And so this is the work in collaboration with Dr. Jeff Rothstein from Hopkins University, in which he generated these iPSC cultures that have the C9 mutation. And the first observation was that that we can actually readily detect using an immunoassay that we developed for PolyGP, was readily detected in the media. And that was an important technical advance, because the fact that not only did it indicate that perhaps a GP is being secreted, but it facilitated this long-term study in culture where we collected the media following treatment of cells at the respective concentrations as shown here. And what we observed is that around 10 to 15 days, we see a 50% reduction in the amount of the PolyGP being generated. And that was quite an important observation, because these ASOs are very effective. Within 1 or 2 days, you can get a significant reduction in the RNA. But the half-life of PolyGP appears to be around that time frame, suggesting that it does take time for this protein to be degraded and turned over, and independent of any type of degradative signal, such as a lysine residue, right? That these proteins presumably do not have. And this is just the lysate showing, confirming at day 20 a reduction inside the cell as well. So we did a series of ASO preclinical studies in our animal model, trying to understand whether or not that this could show efficacy in vivo. And so, as you can see here, in fact, when we injected animals at P0 with a 66 repeat, and then dosed them at 4.5 and allowed them to be treated for 6 weeks with a terminal at 6 months, we saw, in fact, that the RNA, which is not shown here, but the protein is significantly decreased for all the respective dipeptide repeats. But the result that we were really excited about was when we took terminal CSF from these animals and compared them to PBS, the first observation was that we could actually detect the PolyGP in the CSF of these animals. But more importantly was the fact that when the animals that were dosed with the C9 ASO, there was a significant reduction in the amount of the PolyGP in the CSF, suggesting that this might be a potential marker for target engagement for upcoming trials. We since then repeated that study using the larger 149 repeat. We wanted to use this model because of the fact that the phospho-TDP-43 pathology is quite robust compared to the 66. And so what you can see here is at a time point in which we dose the animals with ASO at 3 months, which has quite significant amount of DPR pathology, but the extent of neurodegeneration and phospho-TDP-43 is very minimal at that point. So this is a prevention trial, but understanding that there is significant RNA foci and DPR pathology. And what we observed is that, in fact, there's a significant reduction in the amount of phospho-TDP-43 pathology, as well as this ataxin-2, a well-characterized and marker for stress granules. The animals also, by our UN analysis, showed that there is a protection. These animals show, it's always modest, but it's always significant. There's about a 10% neuronal loss in animals that are injected with the 49 repeat, and compared to the 2R repeat control animals. And those animals that were dosed showed protection. When we looked at the plasma, NFL, neurofilament light, which is a marker for neuronal axonal injury, there were two very gratifying results. One is that the NFL levels in the 149 animals from the plasma showed a significant elevation, very similar to the extent of the neuronal loss and damage that's occurring in the brain. But more importantly is that when we dosed the animals with the C9 ASO, that there was a significant reduction to the 149 PBS, animals. And so we'll see how the human trial, comes into play. I mean, whether or not the preclinical, is giving us any indication of how these ASOs may behave. So working with really an international collaboration, including the UPenn group, in particular, John and David, who were absolutely helpful at providing us with C9 as well as C9 FTD and ALS cases. We wanted to see whether or not we could actually detect this in human CSF. And so using the immunoassay that we developed, you can see that these are all the C9 positive cases that we collected at that time and compared to C9 negative, which would anticipate that there would be very low levels. And in fact, that's what it shows. But we can readily detect, quite a variable amount of the poly-GP protein in human CSF. And when you take this group and divide it into patients who are asymptomatic as well as symptomatic, what you can appreciate just right off the bat is the fact that we can also detect it in the prodromal state. So this result alone, suggested to us that it's highly unlikely that poly-GP would be a prognostic biomarker because of the fact that it can be readily detected in asymptomatic cases. And in fact, doing that analysis, looking at correlations with age of onset and what have you, it confirmed that observation. But I think that this might be a very important point, and also, from a treatment perspective, is recognizing the fact that if we need to treat these patients at an earlier time point, we would have a potential biomarker for target engagement in individuals before the disease can manifest itself and be able to follow it, trying to hit that target as early as possible in truly a prevention trial. The marker that does have some prognostic ability is actually neurofilament light, as well as in what we use in this experiment here is phosphoneurofilament heavy. And they're for all intents and purposes, they behave very, very similarly. The field, I think, believe now is moving towards NFL, for consistency purposes in the field. So what we did was we took the same group of patients that I previously showed where we did the poly-GP study and looked to see, how NFL levels are changed. And what you can see, immediately is that if you look at the C9 positive patients that are asymptomatic, they have very low levels of phosphoneurofilament heavy. But those patients that are symptomatic, with the disease, they have appreciable levels of neurofilament, very, suggestive and consistent with what others have reported for ALS and for FTD and AD. The area under the curve shows, very good specificity and sensitivity to distinguish these groups. But when you break it down, what you really can see is how NFL is able to distinguish patients that have low levels, moderate, or high levels, and their survival rates. And so you can see that C9 patients that have very high levels have the shorter survival compared to low and moderate. So we think that NFL phosphoneurofilament is one biomarker that I think is going to be helpful in how it associates with treatment for any trial, but in particular for the C9 ASO trials, I think is going to be interesting to follow. So we wanted to understand, our C9 149 mouse model showed, prominent TDP-43 pathology, and so we invested some time trying to understand, what is that now mechanistic link? How is that C9 repeat expansion actually causing disease? So we had a couple of experiments that we had in parallel. The first, which I think is really the most exciting in some respects, and that is we were working with Gary Bissell and Wilfred Russell at the time at Emory when Wilfred was there, and now he's at Mayo Clinic. And we became incredibly interested in this other hexanucleotide repeat that's associated with spinal cerebellar ataxia 36. And the reason why that it's so fascinating is the fact that it not only is it a hexanucleotide repeat, but it's nearly identical to the C9 repeat expansion with the exception of one nucleotide. And so you can see here that this is a TG3C2 repeat expansion, and in fact would have, if in fact RAN translation also existed in SCA36, and the genetic mutation is in this gene called NOP56, would have overlapping dipeptide repeats, as shown here for poly-GP and PR. But yet they would have their own distinct, RAN translation products, highlighted here. So we were well poised because, we spent so much time, cloning the C9 repeat expansion that we felt that this would be a relatively straightforward study given our time and investment that we had in working with C9. And in fact, for all intents and purposes, it was. So we took the same strategy that we did for C9, and we made a comparable size construct consisting of 6 repeats for the control or 62 repeats for the SCA36 repeat expansion, and using what we previously made and reported of 66 repeats. So somewhat, imagine that we're doing a head-to-head comparison of 2 RNAs that are remarkably similar, but yet have a very, very different, clinical presentation. One that has obviously an LS FTD and a SCA presentation. So we injected, the 2 cohorts, one with the SCA36 and the C9 repeat, and really want to understand whether or not the behavioral features, the pathological features would be distinct or similar. And so what we observed, immediately in a series of behavioral studies that we have done, including just a simple escape paradigm in which you hang, the animal gently by its tail and see how it, has the ability to escape. You can see that it is significantly compromised and their hind limbs are incredibly weak. And when you look at, time mobile as well as the rotor-rod analysis, that the animals that have the repeat expansion for SCA36 have a very, very different behavioral presentation compared to the C9, which are in the pink and dark purple colors. And so we knew right away that despite expressing these RNAs at comparable level, their RNAs are getting expressed throughout the brain. Already we're seeing a very distinct behavioral feature between these animals. But what became really fascinating is the fact that when we looked in these animals for evidence of neuronal loss, we can see that what we observed was something fascinating. That is, the SCA36, despite having the RNA being expressed throughout the forebrain, had very little neuronal loss. But as I showed you before, that really prominently at 6 months, you can see that there's that 5 to 10% neuronal loss being detected in this animal. This is yet another cohort that we used. It wasn't a control cohort of animals. But when you look in the cerebellum, where the SCA36 presumably would have its greatest impact based on the clinical features of the diseases, there was a significant loss of Purkinje cells only with the SCA36 repeat expansion and not with the C9. And so that selective neuronal degeneration that's associated with it, really indicated to us that although there's only one change in a single nucleotide, that alone is sufficient enough to drive selective neuronal degeneration as well as behavioral phenotypes in these animals. What we also documented was, since we had the antibodies readily available, is whether or not we can detect the poly-GP in the SCA36 animals. And you can see here that there's immunoreactivity for poly-GP, but it was a little bit different than what we found in our C9 animals, which form these very distinct inclusions. It was just more diffuse. And so we were able to identify, now keep in mind that these are incredibly rare mutations with really hotspots in Spain and Japan. And we were fortunate enough to collaborate with a Spanish investigator from Galicia and show in postmortem human tissue that the distribution, can be readily, first of all, detected in these SCA36 patients, but looks remarkably similar to what we find in our 62 repeat SCA36 animals. Moreover, we can show that not only GP was detected in the brains, but also PR, suggesting that RANT translation also can exist in these patients as well. So the most important experiment, to be honest, was whether or not these SCA36, show any evidence of phospho-TDP43 pathology. And so, as I showed you before, the C9 mice have very distinct phospho-TDP43 inclusions, and we just did not see any evidence of phospho-TDP43 abnormalities in the 62, suggesting that, at the RNA level, if RNA had a mechanism of toxicity, and be associated with phospho-TDP43, it presumably is not due to that. Moreover, we looked at ataxin-2, which we previously have shown to show these abnormal accumulation of ataxin-2 as well as other stress granule markers not shown here, but we didn't find any evidence of those features as well. So then, what is it? Then what is really linked to TDP43? And so, we've always been excited and trying to understand that link between, GR, TDP43, and stress granule biology. And so, phospho-TDP43, has been shown, to be associated with TIA as well as other stress granule components. And this is work in our 66 animals, which perhaps can be better detected because of the fact that we're not fixing the tissues, and so we can see things perhaps that can't be readily detected in human postmortem tissue, given the agonal state as well as the long fixing of the human tissues. But we really want to understand whether or not that, could in part GR be the missing link, because of the fact that GR does in fact also induce a stress granule response, not only in cell culture systems, but also in vivo. And that's shown here in the 66 and the 149 where GR and TIA co-localize. So we took a series of cell culture experiments. This one is work from Jim Shorter's lab, led by Hannah, and which he conducted a turbidity and in vitro study, asking the question whether or not if you have GR, which is aggregation prone, but if you throw in TDP43, does it in fact accelerate the combination of the both? And in fact, they do. You can see here that this is TDP43 alone, and when you add in GR, that it greatly accelerates TDP43's aggregation propensity. So is it possible that GR and TDP43 might be interacting? And so we did a series of cell culture experiments here in which we're transfecting GR of 100 repeats, followed by various TDP43 constructs, wild type, as well as ones that are harboring a defective nuclear localization signal that allows it to be expressed in the cytoplasm, one with an RNA binding deficient domain, as well as one that lacks a C-terminal region. And you can see here that the interaction by immunofluorescence clearly shows an interaction between GR and full length, as well as the RNA binding variant, and the construct that only expresses the N-terminal region. So we've done other sets of experiments, including IPs and proximity ligand assays, to really show that there is an interaction between GR and TDP43. What was really important, and I'm not showing here, is that the interaction itself is between the N-terminal region of TDP-43 and not the C-terminus. And we believe that's an important step because once TDP-43 is cleaved and generates a C-terminal fragment, it can spontaneously form an aggregate independent of any other necessary cofactors. And GR is unable to bind to that C-terminal fragment. So that allowed us to then revisit some of our thoughts on how we actually might be detecting TDP-43 and these stress granule markers by looking at an antibody that we generated that has an N-terminal epitope. And so what we've done in this set of experiment is now taking advantage of an AAV-based approach with animals overexpressing GR with 200 repeats, and it's only expressing GR. This is a codon-optimized construct that's ATG dependent. And you can see that these animals have two types of distribution, one that's very diffuse, and one that forms very dense and insoluble inclusions shown here. And both ataxin-2 is capable of recognizing and interacting with both the diffuse as well as the compact inclusions. We can also see TDP-43 interact, but it doesn't interact with the diffuse GR. It seems to be perhaps maybe recruited to more of these more stable structures that not only are part of GR in TDP-43, but the triple labeling studies showed, in fact, that it is a multimeric complex of GR stress granule components as well as TDP-43. And this here we're showing using an N-terminal antibody that I mentioned that that we generated. And you can see it here with the overlay. So we were able to also look at this in postmortem tissue. And as you can see here, GR and EIF3 very similar to our animal model, followed by a TDP-43 labeling, showing a subset of neurons, right? These are not very, very common, but can be detected. And I think that maybe we missed this feature before because most of us have been utilizing either phospho-TDP-43 or the C-terminal antibodies. And so, we can, more readily detect it when we actually use the N-terminal, suggesting that the interaction between the N-terminus of TDP-43 and GR is what initiates this response. So, most of that, once again, has been focused on the C-terminal or the aggregation properties. But, ultimately, as I already mentioned, is that, there's this loss of function that we simply can't ignore. And so there's been a lot of work, that was done in this area. We knew from the earlier days that there's, expression profile changes for RNA targets of TDP-43. But Phil Wong's, paper here really highlighted, for us the significance of that. And so we had a collaboration. This is Aaron Gittler. Many of you know him. And actually refer you to a very interesting commentary that he recently wrote, as well as his very talented graduate student, Rosa Ma. And so the collaboration consisted of really mining Eddie Lee's wonderful data set that he provided, where he took a very different approach compared to what was previously published, and that was separating and identifying RNA transcripts based on nuclear levels of TDP-43. And so you probably are all familiar with Eddie's amazing work shown here. And so Rosa, who's a bioinformatics individual, really mined this data set and using the differential splicing analysis as shown here, came up with a list of targets. And so the ones that previously reported was Stathman-2 and POLDIP3, but UNC13A really stood out for us, and it stood out for us for a number of reasons. The most importantly is the fact that you might have heard UNC13A not only from the work from the biology of Tom Sudoff and the synaptic importance of UNC13A, but from a human perspective is the papers that have shown that UNC13A is a risk factor for ALS, as well as another group that really showed this in a genome-wide meta-analysis, looking at ALS FTD in the various subtypes, and looking how UNC13A reaches significance comparable to C9. So we continue to mind those brains, the brain tissues, and in fact showed in the 7 brains that Eddie had interrogated and provided that in fact that there was an abnormal peak, suggesting that there could be potentially this cryptic exon in which TDP43, based on the binding motif, was between exon 20 and 21. And so work from my lab, from Mercedes Prudencio and Yuka Kyoke, they did a series of post-mortem studies as well as cell culture studies. The first, this is shown here, in which iPS motor neurons, in which we knock down TDP43, show that there's a significant reduction in TDP43 levels, but also in the wild-type transcript as shown here. This is the Western blot analysis showing that there's efficient knockdown of TDP43. But similar to Stathman-2 is that the wild-type transcript is also appreciably lower. So we looked in human brain cases to see if we could actually detect that cryptic exon in a fairly extensive series from Mayo Clinic's Brain Bank, and you can see here that we can readily detect the human transcript in sporadic as well as C9 and progranulin mutation carriers. RNA-seq analysis also, from the New York NYGC publicly available bank allowed us to also confirm this result, mining that RNA sequencing, in which you can see that there are transcripts that are in fact found in areas that are affected with the disease, such as the frontal cortex and the temporal cortex, but were not detected in other brain regions, nor obviously in controls. Bill Seeley, a collaborator, many of us of whom we know at UCSF, did base scope experiments in which he generated a sequence to detect the cryptic exon. And what we can see in human brain, which I think is perhaps more of a clear cut or a visual representation, is that those neurons that have TDP43 pathology and virtually no TDP43 expression, in fact, show the accumulation of those transcripts as shown here, but not in neighboring cells that actually have TDP43. You can see that just shown here are 2 cases. And the controls, obviously, because they don't have the loss of TDP43, do not show that abnormal transcript to accumulate. So there's 2 SNPs, the one that was previously reported, but there was also this other SNP that's also detected, but it's instead of it being in the intron, is actually within the cryptic exon itself. And so we postulate that maybe these SNPs might be part of or rendering an increased susceptibility to the cryptic exon. So to address that, we generated a series of mini gene constructs for UNC13A. This is shown here. This is the reference haplotype, which has the respective sequence. But then we made the risk haplotypes within the cryptic exon, one with the intron itself, and then a mini gene which harbors both of those particular SNPs. And asking the questions, does TDP43 regulate the splicing of UNC13A? And how do those particular genetic variants influence or potentiate, the cryptic exon inclusion? So this is done in cell culture systems in which we treat cells with either, control siRNAs or TDP43. What you can see here is that cells are knocked down, but also then expressing either GFP in red, wild-type TDP-43 or the 5FL mutant, which once again impairs the ability to bind to RNA. And what you can see here with the reference alleles, that when you knock down TDP-43, that there's a significant increase in the amount of the UNC13A cryptic exon to accumulate, which can be rescued when you overexpress wild-type TDP-43. The 5FL seems to be having this potentiating effect, perhaps because since it can't bind to RNA, it's capable of binding to RNA binding proteins and almost acting as a dominant, negative effect. And you can see that, more clearly in the control systems in which TDP-43 is not knocked down, but you get an increase in the transcript. But when you look at the risk alleles, what you see is that the cryptic exon alone is capable of potentiating that effect, which can also be rescued by TDP-43. But the risk allele variant that's within the intron itself seems not to be any different than what you find in wild type, and a combination didn't have an additive or synergistic effect. So what seems to be influencing the splicing the most is the variant within the cryptic exon. So when you look at those SNPs in the human cases that we interrogated and ask, whether or not the number of risk haplotypes can influence survival, we can clearly show that, in fact, that there's quite a dramatic effect. And so you can see here that the individuals that have, both risk haplotypes on both alleles, that their survival probability is significantly shorter than what you would find in individuals with either one or none. So that perhaps is the most compelling human data indicating to us and consistent with the genetic data that confers risk for disease as well as survival. So I think with this group it's very clear that that there's an unmet need for FTD, and our approach has been can we take advantage of perhaps these cryptic peptides, because ultimately at the end of the day what our clinical colleagues or my clinical colleagues and physicians see is a typical case report. In this case here, a woman comes in, behavioral changes, family history, etc., and in the imaging studies support that there is such changes, but what's the underlying neuropathology? Is it an FTD tau or is it an FTD TDP-43? And that's going to be important, especially when therapies become available, as far as how do the physicians guide those patients accordingly, and right now that can't be done. So we've been collaborating with Michael Ward, who's an investigator at NIH, and taking advantage of this loss of nuclear TDP-43, the missplicing that occurs with some of these targets, Stathmin-2, UNC13A. Are there others that might be available that might be good biomarker candidates either at the RNA level, or are these novel peptides sufficient enough if in fact they can be detected and generated? So our strategy has been that, what would make an ideal biomarker, right? You have this abnormal event. If it's a cryptic exon that occurs, hopefully one could be in frame. And that would be the most novel, in which you have the parent peptide with the cryptic exon generating like a novel fusion protein, right? The less ideal would be ones that you have an RNA transcript, such as UNC13A, that immediately follows a premature termination codon. And that would render the RNA to be degraded more readily compared to an RNA transcript that is in frame, or one that actually has a premature poly A tail to it, similar to Stathmin-2, which would render the protein more vulnerable to protein degradation. So taking advantage of that, and in the interest of time, I had to compress a lot of the work that we've done. We actually identified a novel TDP-43 target called MYO18A. There are two variants that do exist, and in fact, when you look at those by qPCR, followed by TDP43 knockdown, both of those variants are elevated, similar to UNC13A, if you will, for variant 1 and 2. And then using antibody-based approaches against that novel epitope, which is in frame and generates a fusion protein, we generated an antibody against that. And what we can see is actually the presence of a very high molecular weight protein, which is consistent with that minus the inclusion of that novel peptide sequence. And it's only detected in cells that have been knocked down by TDP43 and not in the control iPS neurons. And so we believe that this might, at the very least, is a proof of concept that, allows us to reconsider strategies going forward in terms of identifying novel biomarkers for the disease that in this case here would be specific for TDP43. So I won't go over because I think that they're obvious, but as I mentioned, that there may be significant opportunities here, and we're pushing forward on MyO18A as well as other ones, and doing a more comprehensive analysis to really validate whether or not it may be real, looking at human tissues, which we haven't done yet, and we haven't yet looked in biospecimen samples. So in the last just couple of minutes, I just want to end with a side project, but given the timely nature of the pandemic, some of our work that we've done in this space, it emerged in part because many of our observational trials that were being conducted with our clinical colleagues were halted for obvious reasons, and so we asked whether or not we could actually do a potential biomarker study with the number of COVID cases that were coming in into the Mayo Clinic. And so that perhaps was the hardest part of the study, was really coordinating the logistics of acquiring those samples through the hospital and directing them to my lab. And so we were fortunate enough to collect an initial 142 cases. Here, this is just a basic, patient characteristic breakdown of those cases based on race, where we did, have a good representation of, white, Black, Asian, and other in this initial cohort, and some, characteristics of obesity and their length of time and number of NFL measurements that we've conducted during their hospitalization. And so we wanted to ask, whether or not neurofilament, as I already mentioned, a marker well described for neuronal axonal injury, is elevated in these patients. And so compared to controls, this is a log scale, you can see that the NFL serum levels are significantly elevated in COVID-19 hospitalized patients. So much so that when you look at the levels, that nearly 50% of the patients actually have levels higher or 2 standard deviations above the mean. And when you look at 3 standard deviations above the mean, nearly a third of those patients actually have higher levels. So presumably this is not viral driven in the sense that it's a direct impact of the virus getting in the brain. I think that there's reports to suggest that is not the case. But the other complications associated with COVID, respiratory issues and what have you, might be leading to a significant change in NFL and may indicate the potential long-term neurological and cognitive sequelae that will emerge from that. When you look at NFL measurements, whether it's the first or final measurement or the mean, they're all significant. So it really doesn't matter how you slice the pie, so to speak. The NFL levels were found to be significant in multiple time points. Did it associate with any clinical outcomes? And so the points that, or the clinical endpoints that were of interest, to us were, ventilation, ICU, length of hospitalization, and then the modified ranking score, in which, basically 0 is, normal, and, progressively getting worse, ultimately with 6 being passing. And so what you can see here, once we adjusted for age, we were quite rigorous for sex, BMI, and also kidney function. You can see is that the NFL levels did in fact associate with many of the endpoints, including intubation, ICU admission, and perhaps the most importantly is the modified ranking scale, shown here. So, similar to what I showed you for ALS is, does NFL associate with survival with these patients? And so since then, this is, unpublished data, nearly, I think it's around 21 patients that had passed. And so 22 patients had passed. And so what we did was ask whether or not the NFL levels based on a lower or higher value of the 3 standard deviations, does it influence survival? And in fact, what you can see is that very similar to what we found for ALS, that if those patients had very high levels of NFL, their time of death is significantly shorter than those that have lower levels. So we're following this up. Fortunately, we were blessed to get an administrative supplement from NINDS. We will continue now to invite these patients back to follow them up from a behavioral perspective as well as a neuroimaging perspective, and also allow us to look at their NFL levels upon return and looking at other biomarkers, including, for example, tau 217. And so in this very small data set, as we're developing a tau 217 assay, we just wanted to take a peek and see whether or not we see any differences. And so what we did was we chose 15 cases, those that had very low levels, and then patients that had high levels of NFL, and that fairly equal high levels of NFL. And what we found, from this initial study is that actually the phospho-tau have levels that are elevated, but not all of the patients that had high NFL levels had high levels of tau, suggesting that maybe if this is truly a better marker and maybe an indicator of cognitive issues and sequelae, that maybe we might be able to tease out these patients based on their phospho-tau levels. And so much work needs to be done. I just wanted to share this in the interest of collaborations, any ideas or what have you. So obviously this work could not have been accomplished without my wonderful team shown here, as well as the number of really outstanding investigators that I had the privilege of working with, from Emory to MGH, the group from UPenn, and elsewhere. And so I thank you for your time, and I look forward to any questions that you have and any thoughts, whether it's now or later, that you may be inspired with.
Thanks so much, Leonard. That was fantastic. I'm monitoring here the Q&A, so our virtual attendees can feel free to write in a question, and those in the audience as well. I'll be able to go. Kurt, yes.
Fair to say, our readings.
Dave, I can't hear Kurt.
Sure, so I'll repeat Kurt's question. Yeah, okay. Okay, so Kurt's question is it the loss of TDP-43 function or the dipeptide repeat that's causing toxicity, that's causing neuronal loss?
Yeah, that's a great question. I think it's going to be a combination of the both. I think in our animal model, Kurt, you raise an important point that I didn't, mention. That is that while we see phospholipid TDP-43 inclusions, we don't see nuclear clearance yet of TDP-43 in our animal model. And we looked at some of the mouse TDP-43 targets like sortilin, and we don't see, any changes. So, what that would suggest is that, it's a combination of the dipeptide repeats, the RNA, and perhaps even, certainly the cytoplasmic inclusions of TDP-43 that we see. But I don't think that the neuronal loss, that can be attributed to nuclear loss of TDP-43, is part of that. Certainly in humans, there's no doubt, but not in our animal model.
There's a question in the chat from Dr. Lauren Ellman. I can read it. Recent results of tofersen trial suggest that even excellent target engagement may require treatment of pre-symptomatic patients. Identification of these patients in the setting of genetic disease will likely be possible. If an ASO were to be developed, that might be effective in sporadic patients.
So is that more of a comment or a question? The last part is, whether or not ASOs can be developed for sporadic, disease. I think that that's being, pursued. I think that, ataxin-2 is one such, type of, it's not a genetic target, of course, unless you want to consider the intermediate repeats, which is obviously an increased risk for ALS, but that would be such a target that is ASO driven and would have implications for sporadic disease.
We have a question from Eddie Lee. Do we have any handle yet on what drives the variation in CSF DPR levels?
No, Eddie. We don't. I think the fact that we can actually detect that. It's independent of age. We don't know, the true size of these dipeptide repeats, in CSF, meaning that, all we can, measure is just, the relative amounts. And part of that is complex because of the fact that, that there could be a pseudo amplification of poly-GP along the peptide because there's numerous. If you have a relatively large peptide, the antibody will bind to that peptide on numerous, imagine that it's a linear molecule and you have multiple binding sites for the antibody. But I think that's a great question. I think, one that we thought about, and I think that the only one way that one could measure that is by seeing if you can size out, the peptides, at a very gross level, at low, moderate, and high molecular weight.
So Lauren Ellman has a follow-up question. It got cut off in the chat by accident. Could you imagine a biomarker that could be used as a screening tool for a sporadic patient?
Yeah, I think so. I think, some of these TDP-43 cryptic exons, if in fact that they prove to be, successful, would have implications for sporadic TDP-43 proteinopathies. But, much work needs to be done.
And Lauren Ellman follows up that neurofilament might be too nonspecific.
I agree with that.
Leonard, I have a question. The ASO data that you showed in the preclinical model is really exciting, and presumably we would want to turn down the expansion allele. Is there any data on the allele specificity or selectivity of ASOs, or is that something that could be modified in second generation ones as they're developed?
Right. So the ones that we used actually were selected for like the mutant allele. So there is specificity, and I think that Ionis and Biogen have selected one which would target the mutant allele and not the wild type allele. So there is specificity for this one. The Valor study for SOD1, however, was different, and that was targeting both the mutant RNA that would have the SOD1 mutation as well as the wild type allele.
Eddie Lee adds a comment that there's an amazing new technology by Nanopore that allows sequencing of single protein molecules. This could be interesting to use this on DPRs.
Thank you, Eddie.
There's another question from the audience. Okay, so Leonard, the question is, you showed the interaction of GR with the N terminus of TDP43. Is there evidence for the C terminus involvement?
Right, no, there isn't. So GR does not interact with the C terminus of TDP43. We've shown that it's in somewhat supplement data. But as you can imagine, as I mentioned, that TDP43, once it's cleaved, and we've shown that using artificial constructs, it spontaneously can form aggregates, and so there is no need for TDP43 to have any accessory proteins. It can just spontaneously form aggregates in and of itself. And you do find that, of course, Virginia and colleagues have shown the presence of the C-terminal fragment by, urea extractions and what have you.
Well, this was fantastic. We had a very interactive session. Leonard, thank you very much for the talk. It was wonderful. And we'll have a brief break this morning for about 10 minutes, and we'll resume our short talks for this morning on mechanistic work. Thank you. All right, everyone, we're going to resume the morning talk. So the first talk will be by Dr. James Shorter, who's professor of biochemistry and biophysics here at Penn. Protein disaggregases to counter FUS proteinopathies.
Great, thanks so much. Thanks so much to the organizers for the opportunity to present today. And yeah, I'm excited to tell you about some of our latest thoughts on how to counter FUS proteinopathies with protein disaggregators, and I'll explain exactly what I mean by that. So first of all, let me introduce FUS. So FUS is an RNA binding protein with a prion-like domain that is typically found in the nucleus, but in the degenerating neurons of ALS and FTD patients, we find it mislocalized in these cytoplasmic aggregated structures. There we go. And so we see this type of FUS proteinopathy in about 1% of ALS cases and about 9% of FTD cases. And so it's important to note that in the context of ALS, it's typically a mutant form of FUS that's accumulating in these structures, whereas in FTD, it's typically the wild type version of the protein that's getting into trouble and forming these aggregated structures. So our philosophy upon seeing this phenotype in a subset of ALS and FTD patients is, what we'd really like to do is to be able to get FUS out of those aggregates and put it back in the nucleus. So if we could really do that, we'd simultaneously antagonize any toxic gain of function of FUS due to its accumulation in these cytoplasmic aggregates, but also any loss of nuclear function due to its depletion and accumulation in the cytoplasm. So a little bit more information about FUS. So FUS is an RNA binding protein with a prion-like domain connected to the ALS and FTD through both pathology and genetics. Typically a nuclear RNA binding protein, but does shuttle in and out of the nucleus. So there's always a small cytoplasmic pool of FUS. Inside the nucleus, it performs a variety of important functions: pre-mRNA splicing, transcription, DNA repair, RNA transport, and RNA processing, and is really regulating the expression of thousands of human genes. So this loss of FUS from the nucleus is probably an important event in the degeneration of neurons in ALS and FTD. So we see it mislocalized to cytoplasmic aggregated structures in these degenerating neurons, and mutations in FUS are now really connected to ALS. And typically, the mutations in FUS we find in the PYNLS sequence, so the nuclear localization sequence of FUS, which naturally promotes its cytoplasmic accumulation. Some of the other mutations that we find in FUS, we find in what we call a prion-like domain that we see at the N-terminal region of FUS. And it's this domain of FUS that really leaps out to us as very striking as a low complexity domain enriched in only a few amino acids, particularly glutamine, asparagine, serine, and tyrosine. And it gets its name, prion-like domain, because of its similarity to various yeast proteins that are able to form prions, infectious conformations of proteins that have this very same type of domain, which enables them to form prions. And in the context of FUS, this domain of FUS that renders the protein intrinsically aggregation prone and enables FUS to assemble into self-templating fibril structures. And so at the same time, though, the prion-like domain does confer beneficial properties to FUS, so it enables FUS to undergo this process of liquid-liquid phase separation, where FUS will condense from being monodispersed in solution into these liquid droplet structures. And we believe that these are functional membranous organelles. So, for example, if we delete the prion-like domain from FUS, it will be unable to condense on DNA and drive its important function in DNA repair. But at the same time, the accumulation of FUS in these liquid droplet structures places FUS at risk from forming more stable structures. And ultimately, these liquids will convert into gel-like structures and ultimately into the pathological fibrils that we associate with the end stages of disease. Now, what the lab is very interested in, and I should note as well that typically the disease-linked mutations in FUS accelerate this transition from liquid states to solid states. We call this an aberrant phase transition. Now, what the lab is very interested in doing is developing protein disaggregases as a potential mechanism to reverse aberrant phase transition as a therapeutic strategy for ALS and FTD. So what we'd like to be able to do with therapeutic protein disaggregases is essentially reset this phase separation landscape away from solid phase fibrils of FUS and back to monodispersed FUS in solution. Here, we believe FUS could then be transported back to the nucleus where it can go and perform its regular function. So we've developed several ways to do this in the lab, and the one I'm going to focus on today are the nuclear import receptors. I'll tell you a little bit more about those. So the nuclear import receptor I'm going to focus on today, karyoferrin beta-2 or CAP beta-2, also known as transportin, is the nuclear import receptor for FUS, as well as several other prominent RNA binding proteins with prion-like domains. Now, what CAP beta-2 does is it comes and recognizes the NLS sequence of FUS. FUS has a very distinctive NLS sequence called a PY-NLS, comprised of a proline tyrosine motif at its C-terminal end, and this is where it gets its name from, the PY-NLS. What CAP beta-2 does is it comes and binds to the FUS PY-NLS very tightly with nanomolar affinity in the cytoplasm. And this very tight binding of CAP beta-2 to FUS was one of the things that initially made us wonder whether it might also be a protein disaggregase for FUS. The other thing that made us curious is coupled to the biological activity of CAP beta-2. So what CAP beta-2 does is it comes and recognizes FUS in the cytoplasm, engages its NLS sequence, and then has to transport FUS across the nuclear pore channel into the nuclear plasm. And this in itself is a non-trivial task because the nuclear pore complex on its interior is maintained by a dense liquid phase or gel phase of Fg-rich nuclear porin protein that really create a strong selectivity filter that makes it very difficult to cross this nuclear pore complex if you're a protein over a certain size. And so if you are a protein over a certain size like FUS, what you really need to be able to do is bind to a nuclear import receptor. And the nuclear import receptor is very good at crossing this channel by breaking the weak hydrophobic contacts that maintain this gel. And so this ability to bind tightly to FUS and this ability to break weak hydrophobic contacts made us think that CAP beta-2 might be a disaggregase for FUS. And so I should also note that this nuclear transport reaction is regulated by the small GTPase RAN. So in the nucleus, we find RAN in the GT down state, which serves to dissociate CAP beta-2 from FUS, whereas in the cytoplasm, RAN is in the GDP pound state, which permits the interaction between CAP beta-2 and FUS. And so to begin with, we wanted to do a very simple experiment. So here we simply assembled FUS into amyloid-like fibrils in the test tube. And these are very stable structures, so they won't spontaneously dissociate if you dilute them in buffer. And we really think these are bad news. So if we inject these synthetic FUS fibrils into the brain of a transgenic mouse, and we've done this with Don Cleveland's lab and Sandrine de Cruz's lab, these FUS fibrils will elicit an FTD-like phenotype in those mice, causing a protein misfolding cascade of the endogenous FUS and leading to neurodegeneration. So in this sense, we really think that these FUS fibrils are acting in a prion-like manner. Now, what we're going to do in this very simple experiment is simply add cat beta 2 at equimolar concentration with FUS and come back a few minutes later and look to see what's happened. And so if we do that, then we actually very rapidly dissociate these FUS fibrils. So within 10 to 20 minutes, those FUS fibrils have been completely dispersed by cat beta 2. Very striking protein disaggregation activity. We actually don't have anything else that dissociates these FUS fibrils quite as rapidly as cat beta 2. At the same time, this activity made us a little suspicious, so we wanted to do a number of controls to prove that this was a specific activity. And so our first control was to use a mutant form of cat beta 2 that doesn't engage the FUS-PYNLS as tightly as the wild type protein. This form of cat beta 2 is unable to dissociate FUS fibrils. Moreover, if we take a parallel nuclear import receptor machinery, the one that recognizes TDP43, but doesn't recognize the FUS-NLS sequence, this system is also unable to dissociate these FUS fibrils. Next, we were curious to know how this disaggregation might be regulated by the small GTPase RAN. So if we included RAN in the GDB down state, then cat beta 2 could continue to dissociate FUS fibrils, suggesting that this activity would be active in the cytoplasm. But if we included RAN GTP in the reaction, then cat beta 2 was no longer able to dissociate these preformed FUS fibrils, suggesting that once cat beta 2 and FUS arrive in the nucleus, RAN GTP serves to dissociate FUS from cat beta 2. FUS can now go and perform its regular function inside the nucleus. And inside the nucleus, we believe its aggregation is antagonized by binding to its mRNA binding partners. So this was exciting, and we were also able to show that cat beta 2 could disperse liquid droplets formed by FUS. But what about what happens in cells? So here in a yeast model of FUS proteinopathy, when we overexpress FUS, we find that FUS accumulates in cytoplasmic aggregates, and this is very toxic to yeast cells. And in this experiment, what we're going to do is we're simply going to allow those FUS aggregates to accumulate and then switch on cat beta 2. And if we do that, what we see is that cat beta 2 completely disperses the cytoplasmic FUS aggregates and restores FUS back to the nucleus. So boy, would we like to be able to do this in ALS and FTD patients. If we could do this in that context, I think this would be a highly therapeutic activity. By contrast, our negative control, cat beta 2, with 2 tryptophans mutated to alanine that doesn't bind the FUS-PYNLS as tightly, this form of cat beta 2 is unable to disperse these cytoplasmic FUS aggregates. And so I'm just going to tell you that we see very similar results to this in human cell models and also in ALS patient fibroblasts bearing FUS mutations. And also that cat beta 2 is capable of mitigating motor neuron degeneration in fly, expressing an ALS-linked form of FUS. So because most of the ALS cases that present with FUS proteinopathy present with mutant forms of FUS, we were interested to test whether cat beta 2 can also dissociate fibrils formed by those mutant forms, and indeed it can. But there was one annoying exception, and this FUS P525L mutant form of FUS was actually quite refractory to protein disaggregation by cat beta 2. Perhaps explaining why this mutant form of FUS is actually one of the worst mutations in ALS. Patients that have this mutation will develop ALS as young as 10 or 11 years old. And so this lack of activity against this mutant form of FUS annoyed us, and we really wanted to address this and engineer cat beta 2 to be able to recognize this mutant PYNLS that we find in the FUS P525L mutant. And so given that we had a lot of structural information, we're actually able to achieve this through rational design by making complementary mutations in cat beta 2 that restore the affinity of cat beta 2 to FUS-P525L. And so here I'm showing you an experiment where we simply changed 2 amino acids in cat beta 2 to tighten the binding to FUS-P525L. And what we see here is if we assemble FUS-P525L condensates and add wild-type cat beta 2, we're not able to disperse those. But by making those 2 different amino acid changes in each of these engineered variants, then we can now disperse FUS-P525L condensates, whereas the wild type was inactive. So we're excited to take this then to a mouse model. So in collaboration with Neil Schneider's lab at Columbia, what Neil has developed is a knock-in mouse where you express the mouse equivalent of FUS-P525L at the endogenous FUS locus. And these mice come down with motor neuron degeneration, the accumulation of insoluble cytoplasmic mouse FUS-P517L. And to try and treat these mice, then what we're going to do is we're going to inject AAVs that express various forms of cat beta 2 and simply come back 6 months later and ask what has happened in these mice. So the first thing I want to tell you is that simply overexpressing these various forms of cat beta 2 in motor neurons of mouse is not damaging, so that the mice are fine. If we express this dead version of cat beta 2, of course, we don't have any effect on motor neuron degeneration. If we express the wild type version of cat beta 2, we have a mild mitigation of this motor neuron degeneration, but of course we don't expect very strong rescue here as the wild type version isn't very active against FUS-P525L. But very strikingly, if we express one of our engineered versions of cat beta 2 that has better activity against FUS-P525L, then we're able to mitigate this motor neuron degeneration in the context of this mouse model. So I'll close there. And so what we think is going on in disease is that FUS experiences some form of nuclear import defect that allows it to accumulate in the cytoplasm. The neurons then experience some stress, causing FUS to accumulate in these liquid condensates that over time convert into these solid phase aggregates that we associate with the end stages of disease. What we think we can do with cat beta 2 is essentially reverse all of these steps. So we can dissolve the solid phase aggregates, the liquid condensates of FUS, restore FUS back to nucleus. And I think this is something we really want to achieve in ALS and FTD patients presenting with FUS proteinopathy. So cat beta 2 can potently reverse FUS aggregation. It can disperse FUS liquid drops. The final product of that disaggregation reaction is a complex between cat beta 2 and FUS that can be transported to the nucleus and dissociated by RAN GTP. We found that elevating cat beta 2 expression mitigates motor neuron degeneration in my model of ALS FUS. And also we can engineer cat beta 2 to recognize some of the severe mutated forms of FUS that cause early onset forms of ALS. And so this suggests therapeutic strategies for ALS and FTD FUS where we might employ AAVs to deliver nuclear import receptors, or perhaps even find small molecule drugs or other ways to enhance cat beta 2 expression or activity. Could be translated into therapeutics. And here it's important to note at the end that nuclear import receptors tend to be expressed at lower levels in the degenerating neurons of ALS and FTD patients. So I'll stop there. Very grateful for various funding sources, great lab, and a great set of collaborators. Shout out Ling Guo, who's a postdoc who led these studies, who now has her own lab at Thomas Jefferson, and also very talented grad student Charlotte Fair in the lab, who's engineered cat beta 2 to work against some of these ALS-linked FUS mutants. And I'll stop there and thank you for listening.
Very interesting. You have a sense of how acting FUS compensate state fibrils tracking from just one end.
Yeah, great question. So the question is, how does cat beta 2 extract FUS from the fibril state, but also the liquid condensate state? So yeah, so those two forms are maintained by very different interactions, and what that tells us is that cat beta 2 can disrupt both of those different types of interactions. So in the context of the liquid condensate, that is maintained by inter-domain contacts between the prion-like domain and RGG domains of FUS, and also prion-like domain interactions. So we believe those can be broken up. In the context of the fibril, it's always easier to break one off the end, right? Because there's just one contact to break. But this is actually something that we've looked at mechanistically and would be interesting to look into. The point I will make is that FUS, cat beta 2 is a stoichiometric disaggregate, so it has to be there at the same level as the FUS monomer in the fibril. So one cat beta 2 pulls out one FUS. And if you have less than that, then you have less activity. So it is stoichiometric in that sense and not catalytic.
That's the FUS and beta 2.
Yeah. So it's a very tight interaction in the cytoplasm.
Fluorescence to color, and then you can see them interacting in vitro. How they.
Yeah. Right. Yeah. No, we need to do it. I mean, what I'd like to have actually is like an AFM movie of cat beta 2 dissolving a fibril. That would be cool.
It was during the visualization, like FUS was accumulating bright spot within the. That's what you normally see, and is that the normal?
Yeah, good question. So yeah, so in that experiment I showed, it really is just lighting up the whole nucleus. But I guess what we would expect to see, and that's a cell. So in a human cell, what we'd anticipate seeing is FUS also accumulating into some subnuclear organelles as well. They're being diffuse and also accumulating in those structures as well. And that's what we see when we do this experiment in human cells. Great, thanks.
Thank you. That was terrific. Next up is Dr. Sylvia Porta from CNDR in the Department of Pathology and Lab Medicine, talking about heterogeneity of TDP-43.
Good morning, everyone. So today I'm going to talk about the heterogeneity of TDP pathology in human brains and how in the lab we have different approaches and tools to study and understand the meaning of that heterogeneity in FTLD, mainly in other TDP proteinopathies. So here, as nicely Leonard introduced before, TDP protein, it's an RNA binding protein, and it has different domains, and it has nuclear localization and nuclear export signals that although protein is mainly expressed in the nucleus, we can have also that shuttle in the nucleus in cytoplasm, as James told about FUS. So the pathological hallmarks of the TDP pathology was discovered in 2006, and it's mainly the accumulation of insoluble cytoplasmic aggregates in the brain of those patients that you can see that the nuclear protein translocates into the cytoplasm and form those aggregates. And we have that clearance of the nuclear protein. And what, as Leonard was explaining nicely, that it has a loss of function of this protein in the nucleus. So another hallmark of the pathology is the phosphorylation of the protein, and that is the gold standard that we use to follow the TDP pathology in our autopsy cases and also in our animal and cell models is the phosphorylation in the serine 409, 410. And the other characteristic of the pathology is the proteolytic cleavage and fragmentation of the protein. You can see here in the Western blot that the full length is phosphorylated, as you can see for that double band here, but also we observe in the insoluble fraction extracted from the brain of those cases the presence of C-terminal fragments around 25 kilodaltons. So FTD, it's a spectrum of that shares different proteinopathies, as we can have TDP-43 aggregated in the brain, but also tau pathology. And that heterogeneity is not only by the presence of different proteins aggregated, but also you can see here a huge range of different clinical manifestations and also genetic associations with the different FTLD subtypes. So, and also when we talk about TDP neuropathological features, not only those, we have different subtypes that not are only associated to different clinical phenotypes and mutations, but also the distribution of the pathology in cortical layers and the morphology of those aggregates are completely different. For instance, in type A, you can see the cytoplasmic inclusions, but also abundant dystrophic neurites in the superficial areas of the cortex. However, in type B, we have more like a distribution of those compact nuclear cytoplasmic inclusions in the superficial and deep areas of the brain. Whereas in type C, you see those long dystrophic neurites that we call trophy-dystrophic neurites that are all phospho-TDP positive. Finally, we have those type D cases that have intranuclear inclusions that are associated to BCP mutations. And recently, we described with Eddie Lee that new subtype that we believe that it's a characteristic because it has, as you can see in the superficial areas of the cortex, we have those grains in the neuropil that are phosphopositive. And then when we look at the cytoplasm of the neurons, they have a granular staining, but also those GFNs, those granulofilamentous neural inclusions that you can see here surrounding the nucleus of the neuron. So what are those different neuropathological features mean, and are they reflecting maybe the presence of different pathogenic species or what we call also strains? So that's something that we would like to understand. So in 2018, we showed that when we extract insoluble protein from the brain of those FTLD patients and we use them in cell culture and also when inject them into transgenic animal models that express TDP in the cytoplasm and lesser extent in wild-type animals, we show that we were able to induce de novo TDP pathology, as you can see here in the ipsilateral side of the brain, but also most interestingly, over time, that pathology propagates and spreads through the brain to rostral and caudal areas of the mouse brain. We use the same approach to see whether or not those different subtypes have different properties seeding normal TDP into aggregates. So this is just an example of different cases that we are using the study, and here you can see the biochemical characterization, and those cases more or less they look the same. The only thing that we saw is that when we look at the C-terminal fragments and we have those three different fragments, the ratio between the band pattern, it was different between type E and the other subtypes. As you can see here, the lower mobility band, it's more abundant. But other than that, we didn't observe any other difference by Western blot, and the meaning of that difference of the ratio, we still don't know what it means and if it means. So we have that doxycycline-inducible cell model that express a fusion of the nuclear, the cytoplasmic TDP protein fused to the GFP, so we can follow the protein also in living cells. And what we do, we introduce the human material into the cells, and then we wait for 3 days and we analyze the formation of the aggregates using that phospho-TDP antibody. And here, when we were using type A and B extracts, what we observed was the recruitment of the protein into round and spherical aggregates that are highly phosphorylated in the cytoplasm. When we use the type C cases, we were not able to see the formation of the aggregates, meaning that the activity of those subtypes is not as strong as the other ones. And nicely, when we use the type E cases for our transduction, we saw a different morphology of the aggregates that are also phosphorylated in the cytoplasm that we call wisps. So those are fibrillar structures that sometimes are bigger and form like skin-like aggregates that may resemble those wisps that we also see in the type E cases. So here is a higher magnification where you can see the difference in the morphology, but also the ultrastructure. When we analyze the aggregates by electron microscopy, we can see also those fibrillar structures that are different from those amorphous aggregates. So the other nice thing that we saw is that once the brain extracts are introduced in the cells, those can form aggregates in the cells. Here we have the round aggregates that you can see here, that they are really dynamic. They can fuse and form bigger aggregates, like you can see here. And what is interesting is what the cell divide. Those aggregates can be propagated in the offspring cells. And here we can see the cells transduced with type E. We see that this is a really overexposed skin-like structure for you to see the wisps in the cytoplasm. And here you can see when it divides, we can observe the formation of those fibrillar structures in the offspring cells. You can see here, but you can see that the dynamics are completely different other than the morphology of the aggregates. So when we treat those brain extracts with proteinase K, it allows us to see whether or not the conformation of those insoluble proteins are different. And we observe that when we increase the concentration of the PK to digest the extracts, type E had a higher susceptibility of PK digestion for the full length, and also that upper band that characterized the type E. So that suggests that they can have maybe a different conformation and we have different strains. So what happens in vivo? Are those seeds also inducing different morphotypes? So we use that transgenic animal that expressed cytoplasmic TDP, and we inject it into the different subtypes in the hippocampus and the overlay cortex, and we analyze at 1 month post-injection the formation of the pathology. And here, just as an example, you can see that for type A and V, we see a lot of those cytoplasmic aggregates that are also round, and they are all over the cytoplasm. And also what is interesting is that we see a lot of pathology also in neuronal projections, like you can see in the stratum aureus and radiatum. However, nothing in the type C injected animals. And when we look at the type E injected mice, we saw those dot-like and also those fibrillar structures that are completely different from those other type morphotypes. That's also just to show here that resembles that kind of pathology other than the type E, also resemble those wisps that we can find in the motor neurons of ALS patients, that it's been proposed that it's maybe the incipient TDP pathology that will mature over time to form these big aggregates in the neurons. So the other different that those different strains can show us is that when we look at the animals over time, not only we see that type A and B has more pathology in those neurons, but also when we look at the different regions and far from the injection site, we can see that type A and V, they spread and propagate the pathology more than the type E, that it seems to be just localized near to the injection site and really in close areas connected with the injection site. So we believe that maybe the differences in the subcellular localization of those aggregates, it has a meaning in how the pathology spreads in the brain, as we hear a lot of projections that have TDP pathology. Maybe those are easier to transmit from one cell to another and spread faster. So here are the conclusions, but I'm not going to go over, so we have time for the questions. But basically, all our data, the cellular, the biochemical, and in vivo data, is showing us that heterogeneity of the different neuropathological features of TDP may be due to different strains. It's important to understand what are the pathophysiological consequences of having those different aggregates in different subcellular localizations to understand whether or not it has a relation with the different clinical phenotypes in FTLD TDP. And thanks to everyone. And yeah, we have time for questions. I really cannot answer that question because we always inject in one side of the brain, so we see the spreading, but we can see propagation and transmission to the contralateral side. So at least in our model, that's not what is happening. Also, it's not a mutant, so we have cytoplasmic TDP in all the forebrain neurons. So I guess that the model is not the most appropriate to study that kind of like phenomenon with the progranulin. Yeah.
This is very exciting. Are the any differences in toxicity of the different?
Yeah, I check in the cells that we are using. We are using QBI cells. So those are proliferative cells. We check at 3 days post-transduction. So I don't really see any difference in LDH, in viability, and cytotoxicity. So we are planning is like to have a neural model that it will allow us to have more like a physiological consequences of those different aggregates. So yeah, we're working on that. We see pathology in oligodendrocytes with A and B over time. So when we look at 6 and 9 months, and with the type E, we can see some oligodendrocyte pathology also. But I think that also the problem is that as it doesn't spread as the other ones, it's harder to find oligopathology. But if you look at the retrospinal cortex, that is the pathology there, it's really abundant. You can see oligodendrocytes with type E, well, with the extra. And that's interesting because in our model, the oligodendrocytes are not expressing the cytoplasmic TDP. So that means that it's something intrinsic. It's the mouse TDP that it's been recruited into the aggregates. That's really interesting. Yeah, the thing is like the cases that we studied, the first manuscript, those were like FTLD with really short duration of the disease. So what we believe is that maybe that strain is really more aggressive. And that's why maybe that doesn't spread, as Leonard was saying, maybe those cells are dying. So the model that we have now, because it expresses cytoplasmic TDP, it's really aggressive. It has neurodegeneration from the beginning, so it's hard for us also to control what is the consequence of those aggregates. But yeah, that is something that we need to figure.
Thank you, Sylvia. Our next guest is a virtual speaker, Ivy Nguyen, who is currently at Mayo Clinic, Rochester in the Department of Neuropathology, who was formerly in Dr. Eddie Lee's lab, and she'll be presenting on single-cell transcriptomics of AD and TDP43. So this is a video to be shared.
Okay.
Other questions from the group while we're waiting?
Good morning, everyone. My name is Ivy Nguyen. I'm currently an assistant professor at Mayo Clinic, Rochester. And today I'm going to present my talk titled ApoE and TREM2 Regulate Amyloid Responsive Microglia in Alzheimer's Disease. Of note, this was a project that I completed when I was a postdoctoral fellow in Eddie Lee's lab, or the Translational Neuropathology Research Lab at UPenn. So just to jump right into it, shown here is a Manhattan plot, and it demonstrates that recent genome-wide association studies have identified over 30 AD risk genetic loci, many of which appear to be associated with innate immunity. Chief among these risk alleles are ApoE and the E4 allele, as well as a TREM2 R47H variant, which both confer strong genetic risk for Alzheimer's disease. TREM2 is a transmembrane receptor that is highly expressed by microglia in the CNS. It binds to a variety of ligands, including ApoE, and once bound, it triggers an internal activation in microglia. Apolipoprotein, or ApoE, mediates lipid transport and potentially immune function in the CNS. It's mostly expressed in the naive state by astrocytes in the brain, but in disease states has been shown to be upregulated by microglia. So the driving question of this project is, do AD risk genotypes associate with altered microglial phenotypes? To evaluate this, we utilized single nucleus RNA sequencing, or snRNA-seq. To do this, we performed snRNA-seq on 15 postmortem human brain cases, and we took tissue from the middle frontal cortex. After single nuclei suspension, the nuclei were then sequenced using the 10x Genomics platform. We then utilized DESC, which is a novel machine learning-based clustering algorithm developed by Mingyao Li here at Penn, to cluster the single cell populations. And in parallel, 2 independent and blinded neuropathologists assigned an AT score to the tissue. A+ was for the presence of amyloid, and T+ was for the presence of tau. The AT scores, combined with the known ApoE and TREM2 genotypes, allowed us to better contextualize the microglial cell populations. And lastly, we validated our snRNA-seq findings using quantitative digital image analysis in an expanded autopsy cohort. So briefly, DESC is a novel bioinformatics pipeline that projects gene expression profiles to a low-dimensional, nonlinear latent space by an autoencoder network. So this network is then fed into a clustering layer for iterative clustering by a machine learning method. This is where distinct clusters can then be resolved, and this unique approach allowed us to remove batch effect, which is a known limitation of traditional clustering methods. Using DESC, this allowed us to resolve 7 distinct cell populations from these 15 brains that were composed of over 130,000 nuclei or single gene expression profiles. So shown here on the left is a t-SNE plot of the CNS cell populations from these brains, where each dot represents a single nucleus. We used known cell type specific markers to annotate these clusters. And here I'd just like to highlight that the microglial population demonstrated a high expression of tyrosinase BP, which is a very well-known microglial gene. And you can see the subpopulation here in pink. DESC was then used to further subcluster the microglia. And from this, 4 microglia subpopulations emerged, as well as 3 separate contaminant populations. These contaminant populations express additional oligo, astro, or excitatory neuronal genes and may be an artifact of processing. The 4 microglial subpopulations shown here were designated homeostatic, modal, ARM, or dystrophic based upon associated pathology, differentially expressed genes, or gene ontology terms. For example, homeostatic microglia were associated with cases that lacked amyloid and tau pathology, hence the name homeostatic. Another way we designated these clusters was by using differentially expressed genes. So shown here on the right is a volcano plot demonstrating the highest up and down-regulated differentially expressed genes in each subcluster compared to the homeostatic subpopulation. So for dystrophic, for example, ferritin light chain, which is a very well-known IHC marker for dystrophic microglia, was the highest differentially expressed gene in this subpopulation shown here in red. Next, modal microglia were designated due to the number of differentially expressed genes shown here in red that were involved with A, cell motility, B, cytoskeletal rearrangement, or C, extracellular matrix interactions. Last but not least, we then designated amyloid responsive microglia. And we did this by using a few different approaches. For example, we looked at the differentially expressed genes in these ARM versus homeostatic, and we saw that CD163 was the highest differentially expressed gene for this population. We then took this gene and used it as an immunohistochemical marker, a protein marker, to stain Alzheimer's disease tissue. And here we can see that with the staining, we see that the CD163 positive microglia seem to cluster together and label these microglia. When we stained Alzheimer's disease tissue with CD163, we noted many positively staining clustered microglia, which differed from the pan-microglia marker IBA1 staining of the same type of Alzheimer's disease brain. We then used double immunohistochemistry to visualize the CD163 positive microglia shown here in brown were tightly associated with the beta amyloid plaques that were staining in pink, hence the designation amyloid responsive microglia. Once we identified the microglial subpopulations, we wanted to also graph the cell type proportions according to the genotype and/or pathology. So shown here on the leftmost bar graph demonstrates the cell type proportions in the TREM2 wild type cases parsed by pathology, demonstrated in the x-axis. In cases where there were neither amyloid nor tau deposits, we see a high proportion of homeostatic microglia, which is demonstrated here in this light blue bar. However, when you have only amyloid pathology or amyloid no tau, we see a high proportion of the amyloid responsive microglia shown here in this orange color. When you add the tau pathology or A plus T plus, we then see this massive reduction or statistically significant reduction in the ARM microglia shown here in this bar graph. So of note, while the ARM microglia were seen mostly in cases with amyloid pathology, amyloid-only pathology, we do see them in cases with tau and amyloid pathology just markedly reduced. A similar reduction in the ARM proportion was also seen in cases of ApoE4 alleles versus E3 alone. So again, we see this reduction of the ARM proportions shown here in the TREM2 wild type setting. Last but not least, the ARM proportion was also reduced in the TREM2 R47H cases compared to the TREM2 wild type cases. So to evaluate how these subclusters relate to one another or whether they demonstrate discrete transcriptional fates, we utilize trajectory reconstruction and pseudotime analysis. So here is a UMAP projection where, again, each dot represents a single nucleus. And this UMAP projection shows and visualizes how similar the transcriptional profiles of each subpopulation is and how they may be related. For example, here is the homeostatic microglial subpopulation demonstrated here in blue. And we see that it branches and diverges into two distinct activation states, to modal in orange and ARM, the amyloid responsive microglia, in green. We then wanted to validate the single nuclear RNA sequencing data in an expanded autopsy cohort, and we did this utilizing immunohistochemical staining. But to do so, we needed to find candidate marker genes to create these immunohistochemical markers. So what we did was we looked at the different volcano plots for each subpopulation. So again, the ARM versus homeostatic, we saw that CD163 was the highest expressed, differentially expressed gene. For modal, we saw that FGD4 was the highest differentially expressed gene in this subpopulation. And again, for dystrophic, we saw that ferritin light chain was the highest differentially expressed gene in this population. And using these DEGs, we were then able to create or to surmise the IHC markers for the microglial subpopulations in tissue. So all the way on the left, these candidate IHC markers reveal that there are these characteristic microglial morphologies associated with the respective subpopulations. So again, on the far left for reference is IBA1, the pan-microglial marker in a neurologically normal brain. And we see this type of small cell body, ramified and thin processes, demonstrated here that we can also see in a homeostatic population, which we stain with CX3CR1. In a very similar fashion, when we utilize the modal FGD4 immunohistochemical stain, most of the modal microglia demonstrated more of this bipolar or unipolar processes with an enlarged cell body. And in the Alzheimer's disease brain, when we utilize the CD163 and dystrophic staining, we saw that for the R microglia, they had this more amoeboid and clustered morphology with fewer cell processes, blunted cell processes, and the dystrophic microglia had enlarged cell bodies with these beaded types of broken up processes as well. And this is just to remind you also that, again, that CD163 or the R microglia appear to be clustering with plaques. So we see the type of dual staining again. So just to underscore this, we expanded the data that we saw in the single nuclei sequencing cohort and expand that to the autopsy cohort, which included 48 brains of high Alzheimer's disease neuropathologic change cases. We also had them genotyped for TREM2 and ApoE, and then we then stained them for amyloid responsive microglia, CD163, and for beta amyloid. And here we trace the cortices of the middle frontal cortex of these cases, and we calculated the percent area that the staining or cell occupied. We then used these percent areas to create a ratio. And when we created this ratio, it was essentially a ratio of amyloid responsive microglia or CD163 to the amyloid percentage. So again, here in this dot plot, each plot or each dot represents a single case that we stain. And when we plot the ratios of the R microglia and amyloid ratios across the TREM2 wild type setting and ApoE4, we see that there's this marked reduction in the ratios of R microglia to beta amyloid as you move across the ApoE4 genotype. In the same fashion, in the TREM2 R47H background, shown here in these red dots, we see a similar stepwise reduction in the amyloid, the arm to amyloid ratio as you move across the ApoE4 gene alleles. So here's E3E3, E3E4, and E4E4. So this multilinear regression model demonstrates that both the TREM2 and ApoE genotypes were significantly associated with a reduced arm ratio. Another way to look at this is by looking at a plot, a CD163 arm plotted as a function of beta amyloid burden. So again, each dot here represents a single case that we stained for both arm and beta amyloid. And demonstrated here in the background of TREM2 wild type, we're just looking at E3 versus ApoE4 alleles, and we see this significant reduction in the E3 of arm versus of arm in the E3 versus the E4 setting. So this effect was blunted. The same way we saw this effect blunted when you compare the TREM2 wild type cases versus TREM2 R47H cases when you match them for ApoE3 and also for E4. So this is an effect that was uniformly seen across cases that are matched for E3, ApoE3 or ApoE4. So a few conclusions to draw from this study. Utilizing DESC, this novel machine learning cluster algorithm, we were able to resolve microglial subpopulations with distinct transcriptional, pathologic, and genotypic profiles. And that included the CD163 positive amyloid responsive microglia that we termed ARM. This ARM represents a branch of distinct microglial activation state in the setting of Alzheimer's disease that's very specific to beta amyloid deposition. And these 80 genetic risk variants are associated with a very blunted ARM response in human tissue. And the overall fact is that it underscores this diverse and heterogeneous microglial response in human Alzheimer's disease. And the question then becomes, how does this compare to the many single cell or single nuclei sequencing studies that have been performed in mice and in humans? At the time of our publication, we compared our ARM group DEG groups to the different groups that were available. So at the time, there were these different mouse studies that showed these various subpopulations termed activated response microglia, microglial neurodegenerative phenotypes, or the disease-associated microglia. And just very briefly, in the interest of time, we noted that the most common highest DEG expression was in SPP1 or osteopontin, but there wasn't much overlap between our subpopulation of ARM versus the other microglial subpopulations in various mouse models. There have been advancements in the last 5 years where now over 20 studies have utilized this technique or single nuclei RNA sequencing technique various mouse models in human AD brain tissue. In a recent review published by Marco Colonna, he went through and tried to look at these different studies by mouse and by human studies, looking at different subpopulations, their gene signatures. So the gene signatures he focused on was the disease-associated microglia paradigm, and then also the interferon-associated and MHC class 2 associated. Just again, in the interest of time, I won't go through all of this, but this is just to show that even in mouse models, there is this overlap. Each column here represents a different mouse study, essentially in a different AD mouse brain, but it demonstrates that there is some overlap between these mouse signatures and microglia gene signatures in these different mouse models. The same thing can be seen in human AD tissue, but there's less of an overlap, and there's also more diversity in the human AD tissue. Sometimes I think what the discrepancy between some of these results is either by A, technique, or B, the type of mouse model that was used, and C, in human AD tissue, the location and the timing of where they took the tissue to sequence. So ultimately, why do I care? Why do we care? What's so important about microglia? What I tried to show is that microglia are important in Alzheimer's disease. These Alzheimer's disease genetic risk associations largely influence microglia and have this reduction in the ARM response, or a very specific sub-response of microglia. Microglia are complicated. Again, there's this diverse and heterogeneous response that we're seeing that may be associated with the spatial diversity or temporal time-related diversity. So they're a little bit like time lords. And again, just because this is beyond the scope of this talk, I just want to demonstrate that, with this diverse and heterogeneous microglial response, it may change during aging and during disease course, over space and over time. And this has ramifications for clinical trials and future therapeutics. So with that, I'd like to end with just an acknowledgement and a massive thank you to Dr. Eddie Lee, who has been my PI and still continued mentor even while I'm here at Mayo Clinic. I also want to say thank you to Mingyao Li, who provided a lot of the bioinformatics data and has really exceptional postdocs to help with this project too. And just to thank everyone in the TNRL lab. This is an old photo, I know, but I miss everyone back in Philly, and I'm just so grateful for this opportunity. And with that, I'll take questions. Thank you.
Okay, I'll summarize and you can correct me to make sure. So Kurt's question was, in the TREM2 variant, it's clear that there's a depletion of the ARM microglia, but in the ApoE, is it depletion or phenoconversion from an activated to an inactivated or a different state?
Oh, sorry, Dave. I think I lost you for a little bit there. Can you repeat the question?
Sure. You're testing my frontal lobes to retain.
Yeah.
So the question was, so you're showing depletion of the activated microglia in the TREM2 variants, but then in the ApoE genotypes with one or more four, is it a phenoconversion? Is it possible to determine that from a depletion?
Oh, yeah. So we did do staining of just baseline levels of microglia, and they did remain the same. So it may be a phenoconversion, but I guess also the best way to look at that is to double stain and double label these microglia, these ARM microglia, too, in the different states.
Have you looked at these other microglia and other neurodegenerative diseases?
We have, at that time, no, but currently here at Mayo, that is something that I'm currently looking into. I've kind of shifted a little bit of my focus from these are microglia and Alzheimer's disease, but I've shifted it to also looking at normal, quote unquote, normal aging. So we're trying to pair these are microglia with different levels and variations of cognition, and also looking at maybe cognitively resilient populations. So the short answer is kind of yes and no.
That's fantastic because a marker specific for a pathologic microglia is elusive.
Agreed, agreed.
Well, terrific. Great to see you, and thanks for joining us. It's a small world, and good to be in touch. Thank you. So we're going to conclude our morning talks with Dr. Malike Lakamadalia, who is going to be talking about high-resolution microscopy of normal tau and pathogenic tau.
All right, great. So thank you for the opportunity to present our work. I'm going to tell you about a very new direction in my lab today using super resolution microscopy to visualize tau. So I don't need to introduce tau to this audience. This is a microtubule associated protein, as you know, which plays key roles in microtubule stability, microtubule assembly, bundling, as well as regulating motor protein-based intracellular transport. And whenever I find a cartoon picture of tau on microtubules, it always shows tau as very sparsely bound along the microtubule lattice. And this cartoon picture actually contradicts some new in vitro experiments that are emerging. But in fact, we don't really know how tau is distributed on the microtubule lattice. And this knowledge can be important for understanding how tau regulates diverse biological diseases. In addition to its physiological relevance, tau, of course, is pathologically relevant and undergoes aggregation in several tauopathies. And while, again, our tools have been able to visualize and identify these large solid aggregates in intact cells and tissues, visualizing smaller aggregates, in particular these soluble tau oligomers that only contain just a few tau protein, again, in intact cells and tissues hasn't been possible due to their small size. And so this is the challenge that we set out to address. The problem comes from, again, our tools. So here I'm going to show you an IF image of tau in neurons, and here you can see in the zoom up that in the axon, tau appears to be just uniformly distributed everywhere and quite blurry. And it becomes really difficult to discriminate whether tau is on the microtubule and how it's distributed on the microtubule. And this is because the spatial resolution of a light microscope is limited to about 300 nanometers by the wavelength of light, so we can't really visualize anything smaller than that. So to overcome this problem in my lab, we use and further develop super resolution microscopy methods, in particular single molecule based methods, which I will try to explain very briefly with just one slide. So here I'm showing you again a normal light microscope image of microtubules that form a dense network, and it becomes again very difficult to discriminate one microtubule from another within this dense network. And so the trick here is to label the protein or structure of interest with fluorophores that can be switched on and off. And the idea is to switch most of the fluorophores into a dark state so that they're not visible, and switch on just a few fluorophores at a time such that their images are sparse and isolated from one another. So these bluish reddish blobs that you see blinking in this movie are individual fluorophores, And we can identify them because their images are not next to each other and they're not overlapping, they're sparse. And once we identify them, we can also find the position of the fluorophore. The fluorophore is at the center of that blob. And we can pinpoint that very precisely and put a dot to represent the position of each fluorophore. And over time, by activating new fluorophores and collecting more dots, we can reconstruct a high-resolution image, which is no longer limited by diffraction. So again, here's an example with microtubules, where I'm comparing white field fluorescence image to a super resolved image. And I hope you can appreciate that we do improve the spatial resolution dramatically to about 20 to 30 nanometers, so by an order of magnitude. And we can now start discriminating microtubules that are in close proximity. So we set out to apply this method to visualize tau on the microtubule. And to do this, instead of starting with neurons, which are technically a bit more challenging, we started out with engineered cell lines, which were developed in Virginia Lee's lab, and she was very generous sharing these cell lines with us. So we have two cell lines, a cell line that expresses GFP fused to tau. This is the P301L mutant of tau, and this is under Dock's induction. And in the parent cell line, Virginia's lab showed that tau is actually soluble, and it does not aggregate. But after transducing the cell line with in vitro fibrils, Virginia's lab obtained another cell line in which the GFP tau is aggregated. And you can see these big inclusions of tau. And this derived cell line stably propagates those aggregates. So we thought this was nice because we have two cell lines, one that mimics more the physiology or physiological distribution of tau, where tau is not aggregated, and another one that mimics more the pathology, where tau is more aggregated. And so we started out with the parent cell line, and here again I'm showing you just a normal light microscope image of tau. You're looking at GFP signal here in this parent cell line. And I hope you can see that it very much resembles the microtubule images. So it's almost as if we have labeled alpha tubulin, and this is expected because tau is a microtubule-associated protein. It's supposed to bind microtubules. But if we now use super-resolution microscopy to look at tau, instead of getting a very continuous filamentous structure, which is what we see when we use alpha tubulin to label microtubules, we actually see quite a patchy distribution of tau along the microtubule. And we can use image segmentation methods to segment those tau patches, which I pseudo-color coded in the image here. And these tau patches are very small, nano-size. In fact, they're at the limit of our spatial resolution. And we sometimes call them nanoclusters or nanodomains. So the question we had was, what is within these nanoclusters? Because they're resolution limited, we can't really directly see what's inside them. And figuring this out is actually kind of complicated in super-resolution microscopy because even the image of an individual monomeric protein can look like a nanocluster for reasons that I don't have time to go into in this short talk. So we did several additional experiments to address this question, and I'm going to show you a couple of highlights. First, we used T22. This is an antibody that's supposed to recognize tau oligomers rather than monomers. And if we again use T22 to label tau in the cell line, we see a patchy distribution of tau along the microtubules. By the way, we confirmed these are microtubule associated by doing two-color imaging. And then if we compare the T22 images to images where we label all the tau using GFP nanobody, we can do a two-color analysis, colocalization analysis, and we found that this antibody co-localizes with about 35% of the tau nanoclusters, suggesting that there's potentially a proportion of tau oligomers on the microtubule. Again, how many tau proteins are in these tau nanoclusters? To figure this out, we did another experiment where we diluted our labeling such that instead of saturating all the tau in our images, we're sparsely labeling individual tau protein. And this serves as a calibration because it tells us what the signal looks like in our super resolution images coming from an individual tau protein. And we can quantify that signal and compare it to the signal coming from these densely labeled nanoclusters and use that information to calculate what is within the nanoclusters. And this experiment showed us that 60% of these tau nanoclusters are monomers, but about 40% of them are small oligomers containing mainly dimers and trimers. And this is consistent with our T22 experiments, where we find 35% of the nanoclusters co-localizing with T22, which is close to this 40% number. And this was true not just for the P301L tau, but we also had cell lines expressing wild type tau. And we show the same distribution there. It wasn't strongly dependent on expression levels. And finally, we did the same imaging in primary neurons by labeling endogenous tau using a tau-5 antibody. Once again, we find this patchy distribution. And if we do an antibody dilution experiment, we can show that once again, these nanoclusters contain a proportion of monomers, but also small oligomers containing more than one tau protein. What about the aggregated cell lines? So here I'm showing you a representative image coming from the aggregated cell line, and again, the pseudo-color-coded segmentation. You can see it looks quite different from the parent cell line. We still have some microtubule-associated tau, but majority of tau is cytosolic and forms these diverse aggregates. So we still have nanoclusters, but we also have things like small fibrils, more complex structures that resemble to our eye branched fibrils, and more amorphous looking aggregates that grow in size, have different shapes. We first focused our attention on these nanoclusters that we find in the aggregate cell line, and we asked the question of whether they're the same nanoclusters as the parent cell line. And so we did the same dilution and quantification experiments, and interestingly found that, again, we have about 60% monomers, but the proportion of dimers and trimers decreased, and we started seeing higher order oligomers consisting of cordyceps and tau protein. And when we, again, did two-color imaging with the microtubule, we could actually find some of these higher order oligomers associated to the microtubules, so they're not just in the cytosol. And this suggested to us that potentially the dimers and trimers can template the formation of these higher order oligomers. Now, what about these larger aggregates? First, because they're so diverse, we developed a machine learning based shape classification algorithm to classify them into distinct categories. And I'm showing you just a few examples. So again, we have structures that look like linear fibrils, things that resemble branched fibrils that again grow in size into more complex amorphous aggregates. And we asked, what is the nature of tau within these aggregates? So to start looking at this, we also did labeling using 2 phospho-tau antibodies, 3-on-231 and 88, that recognize hyperphosphorylation at different residues. And we obtained super resolution images, again, with these antibodies, which I'm showing you, again, some representative examples. And we noticed that in the 3-on-231 images, we were finding more complex looking aggregates, whereas in the 88 images, it seemed like the aggregates resembled more linear fibril-like structures. And when we quantified this, indeed, we found that the linear fibrils were enriched in the 88 images by about 2 to 3-fold, whereas the more complex branched fibrils were enriched in the 3-on-231 images by about, again, 2 to 3-fold. And this suggested to us that distinct hyperphosphorylation and distinct tau residues can be associated with the formation of morphologically distinct tau aggregates. I'm not going to repeat all the conclusions. I hope I could convince you that using super resolution microscopy, we could get deeper insight, quantitative insight into how tau is distributed along the microtubule as well as tau aggregates. Where are we going with this? So we're really quite excited. This is a new direction for us. But so far, we've done all the imaging in engineered cell lines, and we're excited to take this work into human brain tissue in collaboration with Eddie Lee's lab here at Penn. Here I'm showing you one example from brain tissue with high Alzheimer's disease neuropathologic change, which we labeled again using a phospho-tau antibody. And once again, we find very diverse aggregates in this tissue, which we can first sort by size into small and large structures, and then classify by shape using our machine learning algorithm into circular nanoclusters, linear fibrils, branched fibrils, etc. And this is extremely preliminary, so take this with a grain of salt. I don't really expect you to read this plot at all. But if we compare different phospho-tau antibodies, we again start seeing differences in terms of which structures are enriched and in the images detected by these different phospho-tau antibodies. So we're excited to continue this work. So I will wrap up and thank the people who have done the work. This work was spearheaded by a very talented graduate student in my lab, Melina Giparaki, who did all the work in the engineered cell line that I showed you, and she is now actually continuing this work looking at tau clearance using super resolution microscopy. And she's followed by another very talented graduate student, Adriana Santiago Ruiz, who is taking this into brain tissue. I'm grateful for our funding sources, and in particular, the pilot grant that we received from the Institute on Aging that really pushed this work forward. I'm also grateful to Virginia Lee for providing us with the cell lines, and Eddie Lee for the collaboration on the tissue. And thank you very much for your attention. I'm happy to take questions.
In the literature, I think that it's incorporating the so bind to microtubule, kind of most as they up or down and both directions. And then at the node of RON-VA, and so that's when phosphorylation occurs, because then, it's much narrower at the node of RON. So that, the microtubule has to go through, and the tau has to go through. So the tau then get phosphorylated and fall apart. So how does that model that has been around for years, and jive with your model?
Yeah, great question. So I'm going to repeat the question because I think people online cannot hear it. So the question is whether, the model is that in the neuron, tau is everywhere on the microtubule and then falls off at the node of RON-VA because it's phosphorylated, because the microtubule has to go through this narrow region. So we have, not looked in vivo in neurons. We only looked, in engineered cell lines so far, and in primary neurons that don't have the nodes of RON-VA. So I can't really say whether there is patches of the microtubule at the node of RON-VA where there is absence of tau. But we do see the tau distributed. Right. Right. Yeah.
Very important on the human tissue. You're looking at pollinated axon.
Yes, exactly. So we could potentially address that question with the imaging of the tissue. Yeah, that's a great suggestion. Thank you. Yeah. Oh, that's a great question. So the question is whether the phosphorylation state of tau changes as oligomers mature, or whether the phosphorylation state of the monomer itself determines what kind of aggregates you can get. So this is a question we're very interested in addressing. It's difficult to, go beyond correlation to causation, right? Right now we have just correlation, and we have, very diverse aggregates. We haven't figured out a way of watching the aggregates grow and mature in time to follow them. But the one sort of experiment that we bounced around our heads and was to make tau, phosphomimics of tau, right, with different phosphorylation at different sites, and then induce cell lines with these different phosphomimics and see what types of aggregates result. That could start going towards addressing the question that you're asking. But yeah, it's a very interesting question. I don't have a direct answer yet. Jim.
So,
Yeah, so the question is, the people, especially doing in vitro experiments, have shown that tau forms liquid droplets. There was a recent paper that showed that it forms liquid droplets actually on the microtubule itself, and these droplets regulate, many things. Our data is not consistent with that, at least, in these neurons that we looked at, as well as engineered cell models. What we find are much smaller than these big liquid droplets. And so, I mean, you're in this field, it's very, not so difficult to form these droplets in vitro, but then, the physiological sort of relevance and state is still kind of under debate. We don't find it in the neurons, at least not in the models that we have looked at.
Thank you so much. That concludes a wonderful morning of talks. We have an announcement from Dr. David Woke, Director of the Penn ADRC, about training and funding opportunities, and then a brief announcement about lunch and poster session.
Hi, everyone. I know you're all excited to go to lunch, but I really appreciate Dave giving me a few moments to tell you about some opportunities for our newly funded Alzheimer's Disease Research Center in terms of training opportunities and funding opportunities. I do want to just compliment Dave on putting together just such an excellent morning session, and I think this afternoon promises to be just as good. So the Alzheimer's Disease Research Center has a few different possibilities for additional training. One of them are our developmental grants. The RFA for that recently went out again. These grants are for 1 to 2 years. They're up to $75K. The second year is kind of competitively determined based on progress in the first year. We've tried to actually dovetail these developmental projects with these retreats, and so thematically, things that are talked about today, mechanistic ideas within proteinopathies, as well as translation to human studies, are certainly of interest to us. The general theme of the ADRC is around heterogeneity, kind of writ large, but specifically related to polypathology, phenotypic heterogeneity, as well as ethno-racial heterogeneity, particularly thinking about social and structural determinants of health. And so, again, work around those themes would be of great interest to us. And in terms of a sort of target applicant, we're looking for people who generally are at the assistant professor stage, but also postdoctoral fellows are welcome to apply as well, and a strong grant from a fellow would also be strongly considered. More mid-career individuals, if there's sort of a shift in your interests, also are welcome to apply. I'll be here all day today if people have questions about that particular mechanism. and certainly am welcome by email or other means to chat about any possible proposals. The two other things I just wanted to mention, one is as part of our research education component of the center, we have something called Clark Scholars, which we will help support or supplement funding up to $20K for, again, 1 to 2 years. This is a little bit more targeted, again, towards postdoctoral fellows and also graduate students who are doing work in the ADRD space. And we have a number of mentors within the Clark Scholar program that can serve as either career or scientific mentors, but there's also possibilities of collaborations of people outside that list. So again, if you're at that stage of your career, please do reach out to us. And there, I think, is a brochure out front about how to contact us for that. Contact Cassie, she'll get the emails. And then one other final thing, which may be less relevant to people in this room or online, but maybe something you can help spread the word about. Secretary Torres mentioned that we got this grant from the state, and that grant's focused on African American and Black individuals in the sort of preclinical stages and aging stages, or normal aging versus preclinical disease and Alzheimer's disease. Part of that, though, is a training component, which we've combined with the Alzheimer's Disease Research Center for undergraduates, but some graduate students who want to do a summer internship with us and identify themselves as Black or African American. And we have a number of mentors in that area that can serve that role for a 9-week sort of course and research opportunity, as well as I think a nice stipend for the summer as well. So anyway, I just wanted to introduce you all to those opportunities, and really am open to people reaching out to us and Kathy and others in our group to help facilitate any kinds of proposals any of you have. So thanks for your attention.
Okay, so it's time for lunch. We're going to try to start right at 1 to keep on track for the afternoon sessions. The posters will be visible here as well as online, and if you scan the QC code up front, it'll also be able to load up the posters to review, and we'll go over the poster winners at the end today.
And just a very quick announcement about lunch. We do have box lunches available for all of our speakers and our registered participants. They are in room 102. Because of pandemic guidelines, we're not able to eat indoors as a group, but feel free to pick up your box lunch. You can take it outside. There's seating out in the lobby. There's seating over by the coffee area. We just can't eat as a group. You can't eat in the auditorium. So room 102, you'll find your box lunch. Thank you.
Yeah, my son, he just started preschool, said that they haven't seen kids behave like this before. They talk to each other for weeks, and now they're starting to talk about brother, just kind of talk to other people. It's like, just not so yeah, I think pandemic is development all sorts of things. Yeah. I'm sure my daughter is high school.
Hey, you can get candy, hard to get you to get back in. Yeah. Yeah, that's good. That's good.
I read all. Yeah. I love it. And it's like
Yeah, so it's move. Get rid of shelves and well, yeah. And I
Awesome, so it's all was that. Oh, great. Okay. Oh, like
yeah.
okay. Sorry.
Yeah, so I think that.
Oh, for speakers?
Yeah, that was so good.
Yeah.
Yeah, people ask questions that you
Yeah.
I'll tell you,
See how much control joint have.
Okay, welcome back, everyone. Okay, so we're going to go next to Dr. Fanny Lung in the Genomics Center, who has a talk virtual and is joining us remotely. So we can load that up while we get the rest of Paul's talk loaded for the conclusion of the short talks.
Good afternoon, everyone. First, I would like to thank Dave Irvin for inviting me and organizing this wonderful retreat in this challenging time. And thank you all for joining this event. Today, I'm going to talk about our experience in using high-throughput sequencing technology to identify circulating small RNA signatures in Alzheimer's disease and potentially in related dementias as well. A non-coding RNA is an RNA molecule that is not translated into protein. They function to regulate gene expression at the translational and post-translational level. Small non-coding RNAs are named because they're relatively short, less than 200 nucleotides long. The most well-studied class of small non-coding RNAs, which I believe most of you have heard about, are microRNAs. Other types of small non-coding RNAs include transfer RNAs, small nuclear RNA, small nuclear RNA, POE interacting RNAs, and tRNA-derived fragments. Different classes of small non-coding RNAs reside in different compartments of the cell to perform different roles. They are participating in different biogenesis pathways, serving as post-transcriptional regulators of gene expression, Some are also involved in epigenetic processes, and studies have shown that they play a role in heterochromatin formation, histone modification, DNA methylation targeting, and gene silencing. In recent years, a lot of microRNA discovery work has been performed. This review paper summarized on top of the A-beta hypothesis pathway of Alzheimer's disease pathogenesis, what are the potential microRNAs involved in each of the steps. Example, microRNA 106, which is downregulated in Alzheimer's disease brain samples, is shown to directly binding to APP mRNA, whereas microRNA 146A is selectively upregulated in hippocampus and temporal cortex, which are regions affected by the Alzheimer's disease. But of course, more work is needed to understand in more details what these microRNAs do in disease pathogenesis. This study was published last month by Bart Struper's group. MicroRNA 142 is the microRNA whose expression is the most consistent across more than a dozen reported studies profiling Alzheimer's disease subjects. Through a variety of experiments performed in both adult mouse brain and human adult neural stem cells, they show that microRNA 142 is downregulated in these human adult neural stem cells and is regulating neurogenesis. Also, as the title concludes, restoring its expression in these stem cells rescues memory deficits in Alzheimer's disease, making this microRNA a very promising therapeutic target. Another collection of studies have focused on other classes of non-coding RNAs. These few studies have shown that changes in transfer RNAs, small nuclear RNAs, and PV-interacting RNAs are disrupting different biological mechanisms related to neurodegeneration. However, for biomarker profiling, most Alzheimer's disease biomarker studies reported to date have been performed in serum, plasma, or different regions of the brain that are related to Alzheimer's disease. These studies are able to identify, for example, microRNA 107, whose expression correlates with base1 and nucleic plaques density in temporal cortex in Alzheimer's disease subjects. Others are summarized in this table in this review paper, show that a handful of these play a role in Alzheimer's disease pathophysiology. What's more, the current focus of this studies have been on microRNAs only, one type of small non-coding RNAs. These studies were relatively small in scale. Usually the microRNAs are profiled using real-time quantitative PCR or using some pre-designed microRNA arrays, such as those generated by Tedman. The limitation of using these technologies is that they only focus on microRNAs, not other classes of small non-coding RNAs, which are also shown to be functional in neurodegenerative disease-related mechanisms as shown in the slides earlier. The challenge of the studies has always been scalability and reproducibility, which could be caused by inconsistent way of collecting samples, different RNA extraction protocol used, different study design, and so on and so forth. To address the limitations of these current studies, in this project, our goal is to develop an experimental protocol to profile small non-coding RNAs from fresh frozen cerebrospinal fluid, as we believe this is the closest proxy biodata source to measure what's happening in the brain. These experiments are performed genome-wide, so we are not just limited to small non-coding RNAs that we know of, but also we can profile anything that could get flushed into the cerebrospinal fluid through whatever is happening in the brain. Here is the flow of the project. First, we worked with Steve Arnold when he was still at Penn to collect fresh cerebrospinal fluid from the UPenn Memory Center and then freeze them immediately to minimize RNA degradation. Then we'll extract cell-free small non-coding RNAs using the QIAGEN kit listed here. Next, we perform small RNA sequencing using the Illumina HiC 2500 sequencer and perform some novel bioinformatics analysis to identify what are the differential expressed small non-coding RNAs and what potential regulatory roles they play in Alzheimer's disease. We selected 16 individuals in this pilot cerebrospinal fluid study, 8 cases and 8 controls to perform small RNA sequencing. They are all female individuals of 65 years or older. We also identified few freely available studies which profile small non-coding RNAs genome-wide via sequencing technologies in Alzheimer's disease brains. These studies are of similar sample size as a pilot study and were performed in different regions of the brain as described here. To process the small RNA sequencing data, our lab has developed a computational pipeline to turn the sequencing risk into meaningful peaks, which can help to discover and characterize these known and unknown small non-coding RNAs. This computational pipeline SPAR is publicly available under this URL. In a nutshell, we would like to look for how good the sequencing risk gets piled up into this kind of blocks. On the right here is a schematic diagram showing how these blocks correspond to the hairpin structure of a microRNA. The better the shape is, and the higher the block it is, the truer that the small non-coding RNAs are being expressed. In our pipeline, we first identify the blocks in the discovery stage. Then we overlay the existing annotations onto that in the characterization stage. That's how we discover known and novel small non-coding RNAs. Before doing any analysis, we took a look at the quality of the small RNA sequencing data. Shown in the y-axis of this plot are the number of reads. The blue bar represents the cases, and the orange bar shows the controls. On average, there are 15 million reads per sample. After read mapping, quality control, and all sorts of things, roughly 60% of reads remain for quantification and characterization of small non-coding RNAs. Note that the variability among the cases are bigger than those in controls. Across all the 16 samples that we profiled, we are able to quantify 5 major classes of small non-coding RNAs. Surprisingly, only 31% of this were microRNAs. A similar amount of tRNAs were quantified, followed by small nuclear RNAs, and also PV interacting RNAs. Since there are detection variability across samples, we required a minimum detection limit of 10 reads per small non-coding RNA across all sample sequence. Using this kind of thresholding, more than 1,000 small non-coding RNAs were profiled in this dataset. Next, we took a look to see if for all the small non-coding RNAs detected across all samples in this dataset, how many of those are present per sample in terms of percentage, as shown in the y-axis of this figure, stratified by the diagnosis groups in the x-axis. There are 1 or 2 control samples seen to have a lower detection rate on all the classes of loci, especially for microRNAs and piRNAs, compared to other samples. The variability observed in the control samples in general seem bigger compared to those in cases. On the other hand, between cases and controls, there are no significant differences in the percentage of small non-coding RNAs detected. This is very important as we don't want any possible bias in detection rate or sample-to-sample variability between cases and controls group to affect our differential expression analysis results. Being able to quantify these small non-coding RNAs doesn't mean that they could be potentially useful in disease diagnosis and monitoring. To maximize the discovery potential, we decided to focus on any small non-coding RNA loci that express in more than 80% of the samples. Since we have a limited sample size, we define differential express small non-coding RNAs when the fold change of expression between cases and controls is greater than an absolute value of 2. All the CSF and pre-brain small RNA sequencing data that we talk about have been caught using the same computational pipeline, SPAR, that we described earlier. The table here on the left shows the average number of small non-coding RNA loci caught by each sample, separated by the annotated and unannotated ones. The CSF data, which is what we generated, has a smaller number of annotated loci compared to other brain data sets. However, the amount of unannotated small non-coding RNA loci we discovered is way more than the other data sets, possibly due to the different RNA expression protocol being used, or there could be fundamental differences in compositions of small non-coding RNA classes between CSF and brain. Then using this differential express criteria we just described, the percentage or amount of differential express annotated small non-coding RNA loci is higher than the 2 other brain data sets when we profile that in the CSF. This shows that our overall experimental protocol is working pretty well. For our downstream analysis, we focus on the 162 small non-coding RNAs that are found across all this CSF and brain data sets, of which 36 of them are microRNAs. Here we look at the expression changes for some of these microRNAs. First, 3 of these have never been reported in the literature as SRS disease biomarker. But microRNA 2045' is very highly expressed in the CSF and has fourfold difference between cases and controls. It is found to be downregulated in both CSF and brain datasets. On the other hand, we replicate 5 microRNAs of which the expression changes align well with reported literature based on the same data source example, microRNA 126 5', though it's barely detectable in the control in CSF, is upregulated in the cases. This is also reported in previous literature, and its direction is the same in the brain datasets. microRNA 150 5' is another interesting candidate since it is highly expressed in both cases and controls, downregulated in all the datasets we analyzed, but was reported to be upregulated in other brain studies. The last 7 microRNAs shown at the bottom are the ones that are found in all blood, CSF, and brain. The first in this list, microRNA 425 5', is confidently detected in our AD cases in CSF data, but shown to be downregulated in multiple studies profiling blood and brain. This is a candidate microRNA that's definitely worth further exploration. To understand what the small non-coding RNAs do biologically, we first use the nearest gene approach to look for genes that could be regulated by the differentially expressed microRNAs we identified, and then overlap them with the MIRTA-based list of experimental confirmed target gene list. Listed in this table are the known AD genetic signals obtained from the Lambert et al. Genome-wide Association study paper in 2014, and the potential role of these genes, including microglia-related immune functions, are listed on the right. Lastly, instead of limiting ourselves just to the Alzheimer's disease genes, we performed the pathway enrichment analysis on all the 36 differentially expressed CSF microRNAs we identified in this pilot study. First 11 of these microRNAs are regulating some mRNAs as shown by the MIRTA base. Then out of all these 43 microRNA mRNA pairs, 12 of these mRNA targets are experimentally confirmed ones. Here we didn't show the resulting pathways. Instead, in this table, we show that what possible functional roles do these genes have in Alzheimer's disease, including immune, synapses, as well as aging and cell death. I hope I have convinced you that small non-coding RNAs are no longer dark matters in the human genome. Their roles in different mechanisms involved in Alzheimer's disease and related dementia is becoming clearer. In summary, we successfully developed an experimental protocol to capture and quantify the differences of small non-coding RNA signatures in cerebrospinal fluid of Alzheimer's disease samples. Though the samples were obtained from different individuals between the cerebrospinal fluid and the brain, through our novel bioinformatics analysis, we were able to confirm some of the known signals and identified new circulating candidate small non-coding RNA signatures in our pilot CSF dataset. The microRNAs we identified were found to target few Alzheimer's disease genes reported from Genome-wide Association studies, including MAPT, IMPP5D, etc. Pathway enrichment analysis on these targets have confirmed known and related functional roles with respect to Alzheimer's disease. All in all, these promising results provide a good foundation to further understand the relationship of circulating non-coding RNAs detected in CSF and in plasma or brain, and what regulatory roles they play in disease pathogenesis and how they could potentially be used as a biomarker. With that, I want to thank everyone that is involved in this project, especially Stefano, who was developing this sample banking protocol with me, and Jerry Schoenberg for providing me a bench space. Also, this can't be achieved without the support of the Penn Alzheimer's Disease Core Center Pilot Award. Thank you all for your attention, and I'm happy to take any questions.
Do we have any questions from the audience? I have a question regarding the CSF measurements and how quantitative the microRNA species are that you're measuring. Is it more of a computational approach of a profile that's more of a state, or is there a quantitative aspect to it?
I think each of the microRNA right now is quantified by the number of rays that are actually piled up at those locations in the human genome. So by those different numbers of rays, we can tell that there are statistical differences across the groups samples that we provide.
And you're pairing with brain tissue, so then you could look and see if some of these relate to the degree of tau pathology or plaque or other clinical features like disease duration.
Yeah, so this is kind of like a current limitation of their study because the samples, the brain data that we show is actually not the same brain samples from the CSF samples that we collected. So they're from different individuals. So in the future, we hope that if we can collect this to resources at the same from the same individuals, that definitely will be helpful to understand more questions.
Okay, terrific. Dave Wolk has a question. So Fanny, the question was to please comment on the use of plasma or blood. We have extensive blood-based specimens in the cohort, and could you apply this to those specimens?
Definitely. Actually, profiling in the blood is way easier than profiling in the CSF because of the amount of RNA needed to perform sequencing. But yeah, that's definitely doable.
Okay, well, thanks so much, Fanny. That was great.
Thank you.
Next, we have Paul's talk loaded up, and we can conclude.
Who's coming up first?
So that's it, yes. And you don't have to worry about the
You know it? Is that you? All right, thank you. Oh, one last chore. Sorry about that. Wow, it's magic. Thank you, thank you, thank you. All right, all right. Now okay, so somewhere over here, and talking about the fact that we performed this mapping of tau pathology burden in 3 dimensions. We did it in 18 specimens out of those 15 that did not have FTLD tau pathology. We included in a group-wise analysis, and this is the kind of 3-dimensional statistical map of tangle burden, tau tangle burden that we obtained. So a couple interesting things to highlight here are that there's a pretty strong anterior to posterior gradient that you observe. So there's a lot more tangle burden on average in the anterior hippocampus, anterior medial temporal lobe, amygdala, and stretching into the anterior temporal pole as well, with less happening in the posterior. And what we're very interested in next is to look at what happens to the tau threads. So we didn't look at threads here, we just looked at tangles, and the fact that maybe there's something happening with the threads, maybe in the posterior, which might explain some of the in vivo findings that implicate the posterior network in AD. But overall, this is kind of a nice also complement to in vivo tau studies where, these are maps of tangles. Tau studies give us three-dimensional maps of tau pathology in vivo, and we can look at relationships and see if, this is almost kind of a way of validating some of those measurements as well. Another study I wanted to share with you is, I mentioned this before, this is a group-wise atlas that we constructed from medial temporal lobe samples. And in addition to constructing the atlas, we looked at correlations between thickness of the MTL throughout the region with tau pathology. Now, it's a little bit different here. We didn't use those large three-dimensional maps of tau in this analysis. We used the summary measures of tau as well as TDP pathology in the MTL and correlated with thickness. And in this map that you see here is the association between thickness and tau accounting for age and TDP43 pathology. And we observe clusters where there's a significant relationship that are in the entorhinal region, but also we see it in a subiculum of the hippocampus, and we see a lot of it in this SRLM layer of the hippocampus. Well, this is kind of, it's hard to draw a lot of conclusions on this because it's still small data sets. So we're looking at a data set of 29 brain donors from a mixed dementia cohort. So, we're really hoping we can get more cases into our analysis and look at a more less heterogeneous cohort in terms of diagnosis and perhaps see finer scale relationships than we're able to observe. So I kind of talked a lot about MTL, but I wanted to show a little bit of what we're doing, integrating ex vivo imaging into the PEN ADRC, looking at the whole brain. So the objective is kind of to make these types of measurements relating pathology and brain structure possible to everybody who is part of the ADRC community. And part of that is that, we've been very generously offered to have access to every hemisphere and to scan it with MRI. And so we've been doing this now for several years with Dave Irwin's group and with Dylan Tisdall as well, getting imaging in every hemisphere. And because the hemispheres are much larger and we have a lot more of them long term, we had to adjust the analysis methods as well to make them a little bit more able to handle larger samples, but B, faster throughput. So we cannot do this kind of serial histological sectioning that takes weeks on every hemisphere. So we developed a protocol that we think is a good compromise between throughput and ability to get the kind of measurements we want, and it involves picking a couple dozen anatomical regions. Many of those are regions that are already part of the IOA sampling protocol for diagnosing Alzheimer's disease, but we also added some FTLD relevant regions. And each one of these regions is identified on an MRI scan. And then a semi-automatic method is used to measure thickness. You see that in these green blobs on the left is just illustrating this thickness measurement process. Then we use the MRI to guide the printing of a 3D mold for the hemisphere, which is then slabbed. And then we have a little bit more low-tech 2D mold that gets, it's not a mold, it's just a template that gets printed out, but also is used to know when the slabs are further cut into smaller blocks that are then embedded for histology, where they're coming from relative to the MRI scan. So again, we have this kind of three-dimensional tracking of where every piece of tissue is relative to the MRI. And then these blocks undergo histological processing, immunohistochemistry, and the information all gets kind of mapped, or we're still in the process of this, but getting mapped into the INDDs. So you have thickness measurements, semi-quantitative scores. In the future, we're going to be integrating fully quantitative pathology scores, all co-located. So we're analyzing pathology and structure from the same regions. It's a very early look at this kind of data. These are thickness measurements across 16 anatomical regions in our brain hemispheres. This is looking at 23 cases that are more specific. This is cases on more or less on the AD spectrum, and looking at associations again between here semi-quantitative tau score and thickness in associated regions. So we're looking at ERC here, entorhinal cortex. We're looking at correlation between entorhinal tau and entorhinal thickness, and we're seeing some associations in the right direction. So this is kind of encouraging that this may lead to, as the data set grows, the useful findings between for structure pathology correlations. And finally, this is very exciting recent work from Pulkit Khandelwal in our group, who's been using deep learning segmentation techniques to automatically label the cortex in this ex vivo hemispheres. Now, in vivo, there's a lot of work on all kinds of morphology for the brain, but ex vivo with higher resolution scans, there's been a lot less progress, and it's challenging with higher resolution, sometimes more artifacts, the images and so on, less data on which to train your algorithms. So we're very, very encouraged by these early results where we can actually, get pretty reliable cortical thickness measurements across the entire hemisphere. So, down the road, we're hoping to integrate this again into our processing stream. And, if you're familiar with in vivo MRI free surfer, kind of an ex vivo MRI free surfer equivalent, and all the powerful analysis that enables. So to conclude, ex vivo imaging makes it possible to study effects of neurodegenerative disease on the brain at levels of detail that are impossible with in vivo imaging, but it requires more advanced methods. And there's a lot fewer methodologies so far developed for ex vivo MRI, but I think we're starting to catch up with the in vivo world and the field. Not just what we're doing here, but across the ex vivo imaging and image analysis field. There's been a ton of progress over the last few years. And ultimately, what we hope this allow us to do is to discover signatures of neurodegenerative pathologies that currently cannot be detected in vivo, but with this information that we get from ex vivo pathology correlations, we might begin to discover those signatures. With that, I just want to thank all the members of our group and many groups and everybody here who's not in this picture who has contributed greatly to this work. And thank you, and sorry for the fractured talk.
Well, I know we've talked about that. It would be great to how the process of the tools.
Yeah, so Dave was asking about the progress of, translating some of this to in vivo. So, where we are with that, there are a couple of things I didn't really show this, but one thing that we're doing is using these regions or hotspots that we detect using ex vivo imaging and looking at large studies. Our ADRC cohort, as well as ADNI and other large studies, and looking if we're seeing stronger effects if we focus in on changes in those regions that are identified by the pathology. Now, it has been mixed kind of results. So sometimes we look at like when we looked at the tau blob that we got from this three-dimensional tau map, and we looked at aging, we saw promising strong correlations there. Other times we mapped this hotspot to the in vivo data, and it didn't show stronger effect than, for example, our definition of the BA35 or transentorhinal region that we used, that we obtained using in vivo atlases. So I think we still need to go quite a way to define these effects. So we also, we can look at a correlation between tau and thickness, and we see effects, but we don't have enough power to say this is tau without TDP or tau with TDP, or so detangling these mixed effects, I think, is going to take larger data sets than we currently have been able to analyze. So I think this is something that we need to be doing sort of over really medium term to translate it into in vivo.
So,
Yes,
so is it the reason why we are having difficulty seeing some of the pathology? Is it the difference between extracellular beta amyloid versus intracellular inclusion, like everything else? That is, by imaging, it's difficult to see intracellular pathology in vivo. Whereas, you can do amyloid imaging, right, in vivo, and so that actually provides quite a bit of information, except that so they have the picture, right?
Right. So
we see that quite reliably, but we can't see the intracellular inclusion. Is that because it's intracellular?
So, I'm not an expert on PET, but I think for tau, for example, we cannot distinguish on PET threads from tangles. It's a kind of summary measure of pathology where here we look at the histology, we can look at all different. It's the same thing with amyloid, intracellular, extracellular. Those things can be separated. Now, I have to say, amyloid is the one thing we have looked at the least. I think we've kind of started with tau and then moved into TDP. So we don't have yet tools for quantifying intracellular versus extracellular amyloid on histology slides, but I think there's greater specificity, and then for TDP-43 and alpha-synuclein, there's no PET yet available. So I think the hope is that we can find some signatures that are linked to these pathologies, and if PET comes, when PET comes, that'll be great, but I think it still will be the specificity issue. Okay.
Okay, it's my pleasure to introduce our keynote speaker for this afternoon, Dr. David Bennett. He directs the Rush Alzheimer's Disease Center and Rush University Medical Center in Chicago. He's a principal investigator of the Religious Orders Study, as well as the Rush Memory and Aging Project and several population-based cohorts. His work has been extremely influential in understanding many aspects of aging and cognition, including resilience, mixed pathologies, and more recently, molecular omics of different aspects of aging. Dr. Bennett recently completed a 4.5-year term as a member of the National Advisory Council on the Aging for the National Institute of Aging, and was a recipient of the 2018 Tompkins Prize in PICS Alzheimer's and related dementia. So, David, welcome, and thank you so much for coming.
Thanks for the invitation, Dave, and it's wonderful to be here. It's wonderful to be anywhere, frankly, but it's particularly wonderful to be here back at Penn, where I know so many of you and have watched many of you for a long time, and it's a wonderful, wonderful Alzheimer's Disease Center with really outstanding work, and every time I come back I see new things, so it's exciting. So I was asked to talk about the heterogeneity of age-related pathologies in the community. I want to thank my colleagues at the Rush Alzheimer's Disease Center and my colleagues around the country and in Canada that participated in the work I'm going to share today. I want to acknowledge our funding agencies, in particular our study participants, and unfortunately, I have no relevant disclosures. And really the motivating questions that have driven our work over more than a quarter century is why do older people lose cognition as they age? And how could we find therapeutics to treat and prevent Alzheimer's disease and the related disorders? And so first I'll summarize where we are with two clinical pathologic cohort studies of aging and ADRD. So the first one is the religious disorder study. And I know many of you are familiar with these, so I'll be brief. It started in 1993. We've enrolled more than 1,500 older nuns, priests, and brothers from across the United States. They enroll without known dementia. They agree to annual clinical evaluation. Everyone agrees to brain donation. Subset of about 450 get annual bloods. And then more than 375 have developed incident dementia, over 600 incident mild cognitive impairment. We've done more than 850 brain autopsies around the country. And then the autopsies that are done in Chicago include spinal cord, muscle, nerve, and now more recently fat and bone. Here's where our participants come from. Here's Chicago. We're across the country from California to New York, from central Minnesota to New Orleans and San Antonio. We run around the country mostly for men, but we also have a number of women's groups across the country. And then we have 3 African-American orders and a number of small orders of Latinas in San Antonio. So those are all nuns, priests, and brothers, and there's a lot of really cool things about working with them. But if you know the old Sam Levinson joke, insanity is hereditary, you get it from your children, then we realized we'd have to do a study with lay people. So in 1997, we started a study called the Rush Memory and Aging Project, and we've enrolled more than 2,200 residents from across northeastern Illinois. Here, everyone agrees to annual clinical evaluation. It was very deep clinical and very deep cognitive, but also very deep motor and other phenotypes. Everyone agrees to donate brain, spinal cord, muscle, nerve, and as I mentioned more recently, bone and fat. Everyone agrees to annual blood draw. And we have also about 375 cases of incident dementia, more than 625 cases of incident MCI, and about 1,000 autopsies. We work primarily with contiguous care retirement centers across the Chicago area. Here's Rush down here. This is about 50 miles. This is the Wisconsin border. This is the Indiana border. California is out here, and I'm not quite sure what's between here and California. About a third of the participants we recruit as individual home visits to get lower SES whites and to get racial and ethnic minorities in both studies. We can test in English or Spanish. And there's a large common core of data in both studies. They're conducted by the same people, the same trainer, the same people doing the clinical evaluations, the same pipelines for everything. And so we can efficiently merge the data at the item level. So a lot of what you'll see today is really from both studies. Here's a summary of the design. We enroll people without dementia. We test them every year. We document incident disease. We document a wide range of exposures at baseline. This part of the study has all the bells and whistles of a prospective, analytic, epidemiologic cohort study, except everyone is an organ donor. And so we do get the brain, spinal cord, muscle, nerve at the end. And so within a single study, we can look at risk factors for clinical outcomes. We can look at the biology underlying clinical disease proximate to death. We can look at the biology underlying the trajectories of cognitive change. And I'm not going to talk about it today, but even though you get the brain at the end, certainly for cognition, cognition is the output of the brain. And so with a little mental gymnastics, you can think about what you measure in the brain as actually being the mediator between some exposure variable and loss of cognition. So here's just an example of how people change over time. And so this is a random sample of study participants. On the left side is when they entered the study. On the right side is when they died or when this figure was made and they were truncated. And here age is a metric. And what we're fundamentally interested in is why do some people decline rapidly? Why others not so rapidly? Why some people not at all? Even up, with the 85, 90 age group range. So we also focus mostly on cognitive change. Do lots of things where we talk about Alzheimer's dementia, but fundamentally, Alzheimer's dementia is on a continuum, and this is just kind of illustrates that. The blue line are people that developed Alzheimer's dementia. The green line are people that didn't. And this is a random change point model. You see a random sample of the individual slopes as well. So you can see the people developing Alzheimer's dementia, about 5 years into the study, their rate of cognitive decline increases markedly, but they've been declining pretty rapidly over all the years in the study. As the people who didn't develop dementia, they're declining as well. If you do this with MCI, it looks the same, it's just not as pronounced. Now we're going to go to the end of the study. So this talk isn't going to get so much into the exposures, but let's talk about what happens at the end. Well, here is we're looking at 0 is the time of death, and these are trajectories over multiple years prior to death. And so this thick black line is the average cognitive slope. And again, we're interested, in this case, in things we measure in the brain, and as they relate to person-specific differences in decline. Why are some people declining rapidly, others not so rapidly, some people not at all? And then here are a couple people, even right up into death, that are showing some practice or learning effects. And so we're going to ask first, when we look at different pathologies in the brain, how much of this person-specific variability can we explain? And so here's a number of pathologies. Here's AD, this is with a silver stain. Here's A beta. These are counts. We do counts. We can convert this to pathologic disease. This is A beta. We do image analysis to look at the percent area occupied by A beta. This is tau tangles. We do stereology to count density, amyloid angiopathy. Here's cortical Lewy bodies, TDP-43, hippocampal sclerosis. We have AD, we have a few other neurodegenerative diseases, and then we've got a number of vascular diseases. So atherosclerosis, arteriolar sclerosis, here's a microscopic infarct. Microscopic infarct looks the same, it's just you only can see it under the microscope. So here, relative to this mythical no pathology, when I say no pathology, it really means low pathology, like they don't have pathologic AD, but they can have a little AD. If we did an actual no pathology, there'd be one person there. Hard to get to be 90 with nothing in your brain. Here is AD. So you add AD to that. The intercept is a little bit lower. They're declining faster. You add the other neurodegenerative diseases. Now this is an additive model, so we're adding it to the AD. It drops further. They're declining faster. And then with adding the cerebrovascular diseases, the curve is shifted down. It's a little bit faster, not much. The other thing we can talk about is, well, how many different combinations of these pathologies do we see? And this is where, the headache really starts setting in. Because this is just over 1,000 brains, and we have almost 200 or about 250 different combinations of these pathologies. No one single combination, in this case, pathologic AD, is more than 6% of the cases. No pathology, again, it's not meeting pathologic AD or having anything else. It's still just about 6%. But look here, you have almost 100 people where there's a single individual for that particular combination of pathology, right? And this is what we're trying to treat, because this is how this disease looks out there in the world. It doesn't look like this in your clinic, but it looks like this when you walk out of your clinic and you go into the mall and you look around and you see all those old people. And then you close your eyes and imagine what's in their heads. Well, that's what some of us do to have a good time. It gets more complicated because how much of the cognitive decline any individual pathology explains depends what it happens to be doing co-inhabiting in the brain. So AD, if it's the only thing in the brain, it can account for 100% of what can be explained. We'll get to that in a moment. But it may be as little as 20% if it's in there with a bunch of other diseases. Imagine you're trying to power a clinical trial, beta, all right? In most of these pathologies, you do not know whether they exist or not. Right? Cortical Lewy body is the same. Hippocampal sclerosis the same. TDP the same. You can go through the other ones. They're the same. Okay? And the total variance that can be explained is about 43%. So this is the variance of the person-specific slopes. How much of the individual differences of the person-specific slopes, when we throw all these pathologies in the model, can we account for? And it's less than half. These numbers range because they're intercorrelated slightly, and so it depends which one you put in the model first. If you put AD in the model first, you get a whopping 36%, but if you put it in last, you only get 30%. The other non-AD pathologies, 4 to 10, CVD, 8%, and then you have all this residual decline. Now, I wish we didn't call it residual decline, it's residual change. Remember, not all those people are declining. There's some are state, some actually show some practice effects. If we look at this in terms of how much Alzheimer's dementia could we explain, because that gets around that variability by people not declining or slightly improving. Here's with demographics, the AUC, here's pathologic AD, here's the 7 other pathologies, and then we created a dummy variable for the unaccounted for dementia cases. And we get to about 2/3 of Alzheimer's dementia cases. So these 12 pathologies, or in this case, 8 pathologies, about 1/3 is accounted for by AD. So still big effect. Another 1/3 by the other pathologies, and 1/3, who knows? So if loss of cognition with age is a complex function of multiple pathologies, together they explain about half of cognitive change. There's hundreds of combinations. Each one can explain 20 to 100% of the half of what can be explained. And together they account for about 2/3 of Alzheimer's dementia. And AD is still the elephant in the room, or baby elephant in the room, depending how you think about it. Okay, so now let's talk about the path to personalized medicine for ADRD. I'm going to focus mostly on AD. And so over the last 10 years, we've been building a deep multi-omics, a multi-level omics platform in the brain, to a lesser extent in the blood and the spinal cord. But we have the whole genome, we have several layers of the epigenome, we have the chromosomal organization, the microRNA, the proteome, the metabolome, probably some lipidomes, some other domes, no astrodomes. Then, of course, we have our ADRD trace. So I'm not going to talk about all these layers, but I will talk about a couple of them. The first thing we're going to talk about is RNA co-expression network. So you got about 14,000 transcripts. And that's a lot of transcripts. You could do differential expression, which people do a lot of. We do it too. But we can get this down to 47 co-expression networks. So these are networks where there's genes anywhere from dozens to hundreds that are co-expressed, and then it's a nice data reduction. And here we're looking at these 47 networks. First, we'll focus on the left in terms of beta amyloid and tau tangles and pathologic AD. And here's this module 109 sitting here, which turns out to be of great interest. And I'll show you why. When you put this all into a Bayesian network, and I'm just showing you part of this network, you're interested in the network, in the module that drives a phenotype of interest. In this case, we're looking at A beta, which then drives tau, which then drives cognitive decline. So we're going to look at what's in module 109 that's driving A beta. All right, so here's module 109. It's 390 genes. We pick potentially network driving genes. In talking to my computational people, there's got to be a way to empirically figure out which one are network drivers, but we haven't done that yet. And so we just kind of look at the network and pick things that look like they're highly connected. Realize there's no magic to the way I'm showing this. We could turn this any direction you want. We could flip it upside down, turn it inside out. We could spread things around. We're just trying to show as many nodes as possible. And so we pulled out a number of them. We were able to do some knockdowns in a iPSC model. And you get a couple of knockdowns that alter A beta 42 expression in a cell line with a mutation that generates A beta. That's plexin-1 and IPPL1. So now we have a network that drives A beta. We have a number of genes in that network. We can knock some around, and we can affect A beta. So now we're going to go back to that network, and we're going to measure the protein for some of those genes, in particular, some of the genes that we could modulate in the iPSC model. We were able to do plexin B1. We couldn't do IPPL1. This is selective reaction monitoring for targeted proteomics. And now we're going to look at this in our clinical path data and see whether or not the plexin B1 behaves as expected from both the network modeling and from the ex vivo modeling. And so here, plexin B1, controlling for demographics, is associated with cognitive decline. Everything would predict that its effect on cognitive decline would be through A beta and tau, which means if we control for it, right, we're controlling for mediators, so we should lose the effect of plexin B1. And that's what happens. You put A beta in the model, and it's not significant. You add PHF tau, and you just completely wipe it out. So the effect of plexin B1 can totally be explained by its effects on A beta and tau, consistent with the iPSC and consistent with the co-expression networks and the Bayesian networks. So we've got multi-level omics data, can be used to nominate therapeutic targets for AD. Some of the AD targets can be manipulated ex vivo in a neurons derived from iPSC. We can do targeted proteomics analyses, and we can get models that are consistent with those predictions. But frankly, this still doesn't get us to personalized medicine. Thinking? So think about this. From a complex disease that we cannot biopsy or interrogate in a dish like cancer, what is one potential approach to personalized medicine? So fundamentally, we need to study person-specific differences in a group of human brains in a dish, all right? And we basically took an approach that combines epidemiologic study design with human cell modeling. So epidemiologic study design is fundamentally about understanding person-specific differences. And so why might this work in iPSC? Because the polygenic risk for AD is actually pretty strong. All right, so here's the polygenic risk. It's from the Lambert paper. It drives Alzheimer's dementia, it drives all of our cognitive phenotypes, and it drives all the AD pathologic phenotypes and some of the related things that go along for the ride. And so if you model iPSC, you're basically modeling genomic variability since you've stripped off everything else. So we took our cryopreserved PBMCs and we reprogrammed them to iPSCs, and then we induced them to become neurons and astrocytes and various other things. And we now have about 65 or so iPSC lines. Our first paper was with the first 53. So I'm going to show you some results from 53 different cell lines, from 53 different humans, from 53 different people that were autopsied in our cohorts. Okay. And the first thing we looked at was the relation of the AD polygenic risk score with the spontaneous A beta production in these neurons. And lo and behold, the polygenic risk score is associated with the production of, in this case, A beta 42 over 37 ratio. Okay. Then we looked at the relation of some A beta and tau species, some A beta species with some tau species. So in the brain, the human brain, we see pretty good correlations between A beta and tau, and you see the same thing with the A beta and the tau generated in the dish. Then we took a deep breath and we asked, well, how do these A beta and tau species in the dish correlate with the actual pathology in the brain? And it turns out not so bad, actually pretty good. Actually this good for some of them. So here's neuritic plaques and neurofibrillary tangas from the actual brain, and this is the A beta and tau production, A beta and tau over here, production in the iPSC. Then we took a really deep breath and we asked, what's the relation of these A beta and tau in the dish to cognition? And lo and behold, it was even stronger. And so here's global cognition. This is global cognitive decline. This is level of cognition. This is even the Mini-Mental. And you see proximate to death, and you see many A beta species and the tau species strongly associated with cognition, frankly, almost as strongly as the actual pathology measured in the brain itself. We can get person-specific differences in A beta secretion in INs from iPSC derived from PBMCs of autopsied ROS-MAP participants, and they're related to an AD polygenic risk score, they're related to brain AD pathology, and they're related to cognition and cognitive decline over multiple years prior to death. And so you can imagine when you do compound screens to affect the A beta and the tau in the dish, you look at what genomic diversity is driving response, you generate another set of lines enriched with that genomic diversity of interest, and you redo your experiment, and you're basically going down a road, that we've done in cancer now for the last, 20 years, which is looking to see what particular signatures, in this case, what particular set of genes is responding to any particular drug. Okay, that's great, but really, we need another drug for A beta. And I would say yes. After all, it is the main driver of AD, and the ones we have suck. Not mentioning any names because they all suck. Okay, but we also need to think about doing something completely different. Completely different. So think about it. Eliminating any individual pathology will only prevent a small and variable proportion of MCI and dementia. And is it really wise and practical and affordable to develop biomarkers and cocktails that individually target each pathology? The other thing, and I didn't have time to get into it today, but from a public health point of view, treating this disease once it occurs, all right, is a nightmare. All right, so this is a disease that you actually have to prevent. You have to prevent. What does that mean? It means that throughout human history, up until quite recently, dementia and death are in a horse race. and death won. Won for thousands and thousands and thousands of years, and now, depending, where you live on the planet, dementia wins. And so you have to succeed in pushing off dementia, so death wins once again, not by dying earlier, but by getting dementia later. And to do that, you're then talking about potential biomarkers in cocktails that in the United States would need to be given to 100+ million people at risk, right? And you'd have to do that repeatedly. So let's talk a little bit about resilience. Here's a little cartoon of a ninja brain fighting off plaques and tangles. It was commissioned by me and drawn by my son now 6 years ago. Okay, so here is what we call residual cognitive decline. We regress out the effects of pathologies, and you have the residual leftover slopes. Hard to see, but this is the thick black line that represents the average resilience. And these people are more resilient, they decline slower. These people are less resilient, they decline faster. I do not use resilient and vulnerable because this is a continuum. The same thing, all right? It's just relative to the average person. There's no absolute zero. The only brain that has zero resilience is a dead brain. And, there are things associated with resilience. So, for example, here's early and late life cognitive activity. These are in the model together. The blue is the 50th percentile. 0 is time of death. This is multiple years prior to death. These models control for demographics and pathologies. And if you're more active in early life, you have a slower cognitive decline, regardless of what pathology is in your brain. And if you have more activity in late life, you have a slower cognitive decline, controlling for early life, controlling for pathologies. So cognitive activity doesn't much care about what brain pathology you have, it's just protective. Here's some proteins from the snare complex in the presynaptic terminal, complexin 1, also associated with a slower rate of cognitive decline. I'm sorry for this crossing here. This should not go out to -12. This is a SNAP25 syntaxin interaction. We pull down the SNAP25, and then we use that to capture the syntaxin. And so you're looking at the complex protein, so that's also associated with a slower rate of cognitive decline, controlling for pathologies. Here are tyrosine hydroxylase positive neurons in the locus ceruleus. Same thing, more neurons, a slower cognitive decline, control for demographics and neuropathologies. So we've loss of cognition with age. It's a complex function of multiple brain pathologies now that interact with or add to multiple resilience markers, or multi-resilience markers offset some of it. So you get this complicated thing going on, that all results in how much cognitive decline any particular individual has. Now, resilience markers, as I've demonstrated here, exert their effects agnostic to brain pathologies. I will say some, not the ones I've shown you, but some that I haven't shown you do have interactions with brain pathologies, but they're not directly related to brain pathologies. There's no evolutionary pressure to develop separate resilience systems to combat each late life pathology. Okay, whatever resilience systems are in our brain were honed while humans roamed the savannas of East Africa. Okay? It had nothing to do with age-related neurodegenerative diseases. All right, and so much like our immune system falls into two large buckets with a lot of details around them, and they were developed many, many years ago. But by design, all right, our immune systems, by design, can take care of things they've never seen before. HIV, Ebola, Zika, COVID. So the resilient systems in the brain are probably the same in the sense that they were developed around the things that affected people back then. Injuries, animal bites, things along that line, but certainly not late life neurodegenerative diseases, but they were built in a way that they could respond to things that they haven't seen. So the question becomes, can resilience serve as a therapeutic target? So there's a couple of ways we can do this. This is a proteome-wide association study of residual cognitive decline. We call this a Chicago plot. Unlike a Manhattan plot, which drives me nuts, the Manhattan plot shows you the p-value. This shows you the p-value, but the direction of the effect. So these 7 are all associated with greater resilience, and this little guy down here is associated with less resilience. And if you've ever been in Chicago and you go out to on the water or to the planetarium and you look back at the right time, you see the reflection of the beautiful lakefront on the beautiful Lake Michigan. Some of the RNA transcripts for those proteins are also associated with resilience. So there's 8 proteins associated with resilience. We could also pull some of these out of our co-expression networks. So here we're back to our co-expression networks, 47 networks, and now we'll focus on cognitive decline and clinical AD. And here's our friend, module 109, all right, really strongly related to cognitive decline and to clinical diagnosis. And so what's in module 109 that's so good for cognitive decline? Well, if we go back to our Bayesian network, we talked about this little guy over here to amyloid. What's this big, thick, fat guy straight to cognitive decline? Now, realize that the length of the line is meaningless, but the thickness of the line is not. The thickness of the line is related to the strength of the association. So the effect of module 109 directly on cognitive decline is actually much, much stronger than its effect on A beta. So let's go back into that network, 390 genes, in this case 390 proteins, and let's measure some potential drivers of cognitive decline. And so here's a bunch. We talked about plexin B1 already, but here's a bunch of other ones. Now, we're not looking for the strongest, like 10 to minus 16th for IGF-BP5. What we're looking for are significant proteins associated with cognitive decline that are not associated with brain pathologies. So here's 2. AK4 is not related to any brain pathology. ITPK1, has this, minuscule association with PHF-tau that doesn't, come close to withstanding multiple comparisons. Okay, and then we get the different direction of the association. And here we're looking at cognitive decline. Here's 0 again, time of death. These are years prior to death. These are control for demographics and for the neuropathologies. And here's AK4. And what we see here is that a high AK4 is associated with less resilience, faster cognitive decline. ITPK1, a high ITPK1 is associated with more resilience or a slower cognitive decline. All right, so we pulled this out of the same network, but because we use the full continuum of this residual cognitive decline, all right, we can get proteins that drive this either direction, and then of course you would need to target these and try and move them in different directions. So you would try and knock down AK4 and you try and back up ITPK1. AK4, we can make an image of it. I sat through a bunch of meetings yesterday looking at imaging pictures, and I said, I got to get a couple images into my talk or I'm going to be like the real odd guy out. And so here's an image. This is the association of AK4 with brain volume. This is ex vivo voxel-wise deformation-based morphometry. And so basically what we're looking at is how much warping you need to do for each one of these images to get it to the reference template, all right? And depending the direction of the warping, all right, you're going to get whether or not that region is bigger or smaller. And in this case, AK4, remember, which is bad for you, is associated with less brain volume in these regions here. We can also make a polygenic risk for residual cognitive decline. So some of you may see where this is going. If we get a strong polygenic risk for cognitive decline, maybe we can study resilience in a dish. And so this polygenic risk explains about 20% of the variance of decline. Okay, just remember controlling for all the pathologies and the demographics, okay, which is not bad. We can also get a footprint here. This is actually in vivo images with the polygenic risk. We get several different, four different signatures across different brain regions, mostly in the temporal lobe. So again, we got our cryopreserved PBMCs. We generate neurons and astrocytes. And in this case, we're going to generate some proteomic data as well. Okay, so this is the same 53 lines I just showed you. Okay, but now I'm going to show you the heterogeneity of the underlying pathology in these 53 lines. Okay, it's hard to read this, so I blew it up over here. This is just the first column. So we got several different pathologies in there. You can see, there's a lot of pathologic AD, not everybody, but then a smattering of all these other different pathologies, very much like what we see in the humans, but in this case, it's what we see in the humans from whom we generated these 53 lines. And so you can imagine that if we could target resilience, all right, we'd be offsetting the effect of all of these in any combination. So first we look at induced neuron AK4 in relation to the brain pathologies, and we get nothing, all right, which is what we're hoping for because that's what we got with the actual pathologies in the brain with the AK4 measured from the brain. These are the same pathologies, but this is AK4 from the induced neurons, and this is AK4 from the brain itself. And then we looked at a regression with residual cognitive decline, and lo and behold, higher AK4 is associated with a faster rate of cognitive decline, residual cognitive decline. So that's our measure of lower resilience, all right? And it looks awfully like what we saw when we measured brain AK4. So this is induced neuron AK4, all right, with clinical and path data from the human from whom this was generated. And this is from the brain AK4, just showing that it looks the same. We got multi-level omic data can be used to nominate therapeutic targets for resilience. You can get person-specific differences in resilience, proteins associated with cognitive decline from human-induced neurons from the same humans. These associations are probably driven by the person-specific differences in the genomic risk of resilience. They would kind of have to be because that's all you got in those iPSC lines. All right, and then we can do the same thing with future compound screens. All right, if you did compound screens and found something that affected AK4, it would affect it in some lines, but not others. You would look back at the genomic diversity and then try and enrich your sample using another set of lines and rinse and repeat. And that's my talk. We have a lot of data, a lot of biospecimens, and my time horizons are getting shorter, just shorter. And so we already collaborate with a number of people here. We have for a long time. I know Stephen is not here anymore, but got my first R01 with Stephen back in 1998. But some of you weren't born yet. And so, so here's our website for our hub. If you're interested in some of the data, if you're interested in some of the biospecimens, You go in there, it's all documented, it's designed to make it easy to figure out what you're looking at. For a bunch of things, you can figure out how much we have. There's data use agreements there. There's already data use agreements with Penn, so that's easy. Data move easily and rapidly. Biospecimens slower. So let me stop there, and that leaves us time for questions. Questions? Yes. You know, we'd have to see what that compound was. You know, for a lot of these decisions, they come down to risk, cost, benefit, risk benefit, and so assuming it's safe and it's cheap. You know, I'd start with, safe and it's cheap, all right? You can do a bigger study, you can tolerate a smaller effect size, and you might want to give it to people who, are at risk, meaning older, all right, with maybe some memory complaints, not at risk the usual way we do it with genomic variants and stuff like that, but something that is going to portend decline, because you need decline in order to see whether your drug works, right? If it's toxic and expensive, it moves you closer to people who are more symptomatic because you need a bigger effect, and you need it sooner. So there's a lot on the table that dictate that. Yeah, so biologically, this is an opinion because none of this has been proven, but I can see delaying the onset of dementia, delaying the onset of cognitive impairment. You might be able to slow dementia a little bit. You're not going to be able to reverse it. The brain is just a pile of crap by the time you're demented, all right? There's no, your brain is more resilient than any other system in the body, okay? The last thing it wants is to be demented, and it does whatever it can. So by the time you get there, all right, there's just nothing to work with. All right, and so I would want to go as early as possible. Dave?
Great. Market. That's this, but sure.
Yeah, so I showed you a couple little things, so the tyrosine hydroxylase positive neurons in the locus, presynaptic proteins. We have done some stuff with imaging.
That, I guess, leads to greater neuronal.
Well, for the TH positive neurons, that's exactly what it is. It's more neurons, right? Whether you started there or whether something you did help maintain them, or, I don't know. But you can get a density measure there that reflects, the total number pretty easily. But you don't have the total number. Scaling total numbers in a particular region of neurons, is a daunting task. And I think I was talking to maybe Paul about this yesterday. I think it was Paul. but a lot of talks yesterday. But I'm trying to figure out some way that we could combine volumes from imaging with density measures of different regions to estimate total numbers, all right, for either the hippocampus or for other brain areas that you can, circumscribe. There's got to be a way to do that. I just don't know what it is. The math isn't obvious to me, but it seems doable.
I guess the thing I was start off with more normal. Same amount of.
So that's certainly possible, and it's undoubtedly part of the picture. If the way we do epidemiology now is a life course model where you think about early life associations with peak performance. Starting at a higher peak is better, and then separately, what factors are predicting faster change from that peak, right? And they need not be the same, and they're often not the same, and they're both of interest. Except here, we're actually showing proteins. So then, you would have to posit that those proteins are related to those exposures, all right? Because we're not talking about neurons. I mean, I did with the TH, but these are proteins from the brain, and they're proteins from baby neurons. And so, we are looking at these proteins in relation to a lot of our experiential and psychological exposures. If I had something really cool to show you, I would have shown you something really cool in that regard. But we're still at the beginning of that road. So maybe that cool thing that I can't show today, I'll have next time I come to Philadelphia. Yes, sir.
To build on concept of resilience, other fields,
right?
And brain resilience.
Yes. I mean, so in particular, is immune senescence and immune exhaustion related? That's a great question. We do save cryopreserved PBMCs every year. We make our cell lines out of them. The methods to measure immune senescence and immune exhaustion are still eat up too many white cells for me to, eagerly give out those PBMCs, which are worth more than their weight in platinum or catalytic converters or whatever, everybody's stealing from the cars nowadays. That's a great question. It dovetails nicely with all the SNPs in the immune system that are floating out there. Yeah, that's looking forward to being able to do that one day. Mm-hmm.
For influenza,
Mm-hmm.
And we were looking.
Right.
Nobody back then was names of these.
Right.
But I think
there's been little studies, but they're very, they eat up a lot of white cells. All right, and so they're just too precious to part with.
Yeah, eventually. late line depression and sort of in doing research on
Right, right. And depression is another, depressive symptoms anyways is another exposure factor associated with cognitive decline. It's not related to any brain pathology. We have most of the experiential, many of the psychological factors, we really don't care about the pathology in the brain. Some of them that are related to heart disease are related to cerebrovascular disease, but they're pretty weak. And I didn't show it here, but many things that are associated with resilience also have associations with pathology, so they're pleiotropic. So right. Yeah, we looked at vascular depression. We didn't see it, but it was a long time ago. So we have a lot more data now to do it. We should probably repeat those analyses.
this is really exciting because it's thinking that there are and actually therapeutic.
You don't have to convince me it's exciting.
So thank you.
Yeah, no, but people talk about personalized medicine, and frankly, first thing I've ever seen that actually, is a potential path there, because I don't know what else is going to get to individual decision making about any particular drug for a disease in the brain. Whether this all works out that way, I don't know. I will say that these iPSC lines, we are generating more. We just got funded to do 57 lines in autopsied African Americans from these cohorts. It's a little bit more complicated, but you basically, you can do ancestry in a dish as well. So you can, find drugs that work in European, and then you can look to see if they work in African or almost all the Africans in the United States. African Americans are mixed, so they'll work in some and not others, and depends where their genomic diversity is between Europe and Africa. So, and we're looking for ways to generate more lines. If it was up to me, I'd have one for every person. There's people interested in particular genes, so we've identified which lines have TREM2, for example, because somebody was interested in that. They're at New York Stem Cell Foundation, and they're shareable. If you want them, you don't go to the New York Stem Cell Foundation, you go here, all right? And then if you're approved, we let them know. Any other questions? Many of you have my email. I have a meeting at the airport, so I need to leave. But if you have any other questions or anything else, just let me know. Invite me back anytime. Okay, I'm gonna go home today and see my wife and my dogs. We're empty nesters. I'm not gonna see my kids. But take care, everybody. Great to see you.
Thanks so much, David. That was fantastic. We hope you have a safe trip home, and it was wonderful to have in-person lecture. So we reshuffled things a little bit. I think we could have. So I think we'll do one more talk before our break, and then. So this is Dr. Catherine Cousins from the FTD Center, who has been doing a lot of work on biofluid biomarkers and Alzheimer's disease and related disorders.
Thanks for having me. I look forward to following that talk. Yeah, so today I'm going to be talking about strategies that we can use to improve the ability of biofluids to capture the full pathologic spectrum of Alzheimer's disease and related diseases. I'll be talking mostly about cerebrospinal fluid, but I'll talk a little bit about plasma at the end. Oh no, my slides are bigger than the screen. So biomarkers and CSF, along with neuroimaging, have been part of this important progression from Alzheimer's disease that was a condition that was clinically diagnosed to one and with pathology only confirmed at autopsy, to the present recommendation for a biological definition of Alzheimer's based in biomarkers of pathological hallmarks, amyloid plaques, and tau tangles. And one of the focuses of my talk, and David did this nicely, was talking a lot about the heterogeneity. And while we have many advancements in biomarker diagnostics, there's still a need for autopsy confirmation at the end to capture the full pathological spectrum, ADRD. Man, my slides did not translate. Okay. So the ability for us to progress to this biological definition of Alzheimer's disease was made possible by a biofluid signature of that was discovered. That is, this was going to be a graph that showed that phosphorylated tau is elevated in Alzheimer's disease compared to controls and other neurodegenerative conditions. And this was also a graph showing that A beta levels in the CSF lower in Alzheimer's disease compared to other neurological conditions. And so when we combine those two, this yields excellent discrimination of AD from non-AD. As shown here in this autopsy cohort, this ROC curve shows that the p-tau to A-beta ratio in orange has like excellent specificity and sensitivity when discriminating pure AD from FTLD. Oh.
I think it's the translation. You did these on a Mac, and there's nothing he can do.
That's okay.
Okay, sorry about that.
Use your imaginations to picture something more beautiful. But
I'm not sure that.
yeah, I mean, either way, would you guys prefer if I like made a PDF and we came back to this, or do you want to continue to look at?
We can try and troubleshoot and take a break now, and then
okay.
Emily Bargen.
That sounds good.
And we can see if we can improve it.
Okay, so we'll take a 10-minute break. Okay, so we're going to resume now for the rest of our afternoon's talks on translational work and ethics, and Dr. Katie Catherine Cousins is going to resume her talk on biofluid biomarkers and ADRD.
All right, so for last here, I was showing you how the p-tau to A-beta ratio has excellent discrimination for AD here compared to non-AD neurodegenerative disease FTLD, or frontotemporal lobar degeneration. But this binary strategy of positive AD or not AD can't capture all of Alzheimer's disease. So this is a study from CNDR And these are all Alzheimer's disease patients. And this point was really nicely made previous by David, but we have a minority of patients with pure Alzheimer's, with pure amyloid and tau pathology, but a good, a good proportion of them have co-occurring alpha-synuclein, TDP43 pathology, or all four, A beta, tau, alpha-synuclein, and TDP. And so not only are mixed pathologies the norm, I jumped ahead. So this is a community-based study, which also shows nicely that while Alzheimer's disease may be the most common of the proteinopathies, pure forms of AD may be less frequent than mixed forms. So they show this both in white Americans, where pure AD is still less frequent than mixed pathologies, and this may be even more true when we look at non-white populations. And particularly in African Americans, we may see that there's a high proportion with co-occurring Lewy body disease. So not only are mixed pathologies the norm, they're clinically relevant. Here is a study showing hypothesized trajectories. And if we have cases with 3 or 4 proteins, that they have much worse clinical outcomes and faster cognitive decline than monoproteinopathies like tau alone or dual proteinopathies like AD. So an alternative to these binary strategies of just simply AD or not AD is the 2018 ATN framework, giving us an alternative strategy. So here, the ATN framework says, let's combine the 3 core biomarkers, beta, p-tau, and t-tau, into an ATM profile that is along a continuum of Alzheimer's disease. And for this, what you need are low A beta levels and high p-tau to meet a diagnosis of Alzheimer's disease. But unlike binary strategies, what we can now do is potentially or theoretically stratify non-AD pathology. So in biomarkers, this would be called SNAP, the non-AD pathology, from normal patients who are negative for all 3 abnormalities. And then this also theoretically allows us to detect patients with co-occurring pathology, so concomitant Alzheimer's and SNAP. And while theoretically grounded, in practice, it could do better. So this is a study of mine looking at how ATN performs in Alzheimer's disease patients, those with alpha-synuclein positive Lewy bodies, and those with co-occurring AD and alpha-synuclein. And so you can see that a large proportion meet the, under ATN, meet the diagnosis of Alzheimer's disease, and many are on the AD continuum, and only a few are obviously misdiagnosed as SNAP. But when we look at Lewy body patients, now we see the failure to capture these patients. So a large majority are classified either as normal or amyloid positive alone. Under a quarter are classified correctly as SNAP. And when we look at those with co-occurring disease, so alpha-synuclein and AD, we see that a smaller proportion meet the definition of Alzheimer's disease. More of them are classified either as normal or amyloid positive alone. And this is curious. A similar proportion are classified as Alzheimer's change plus SNAP as the AD group. So it's not clear that this is a meaningful designation. And if I compare this to the t-tau to A-beta ratio, a binary, this is less detailed, it's less ambitious, it's meeting what it claims to do. So patients who are positive for AD at autopsy, AD, and those with co-occurring AD and alpha-synuclein are positive by the t-tau to A-beta ratio, and largely those who are absent of AD show that they're negative. And I guess this is all to say that there's room for improvement, and the ATN system was designed to be improved upon. So I think we can meet that challenge by tackling two of the limitations of current biofluid-based strategies. The first I'll talk about is detection of non-Alzheimer's disease pathologies. And I think the poor detection of non-AD with the ATN framework has a little bit to do with why N status is in parentheses, and that's because it's framed as optional. It is not required for a diagnosis of Alzheimer's, only a status are. And so it's a little bit ignored, but if you are interested in detecting non-AD pathologies, then it's pretty vital. For detecting both SNAP, many of the cases our status is useful, and it is necessary to potentially detect these patients, those with both Alzheimer's and SNAP. I think the other issue is that the current recommendation for a marker of neurodegeneration, which is t-tau, high levels of t-tau, has two problems. First of all, t-tau is highly correlated with p-tau, and so the two biomarkers are not orthogonal to each other, and so it's not giving different information. And the second is that p-tau, although framed as a non-specific marker of neurodegeneration, is actually lower in non-AD proteinopathies. It's less good at detecting neurodegeneration in those other conditions. So I showed you this before. ATN doesn't well classify Lewy body disease patients, and here again I'm going to show you that it doesn't well classify patients with FTLD pathology. Yeah, so this skipped. But what I want to highlight here is, so here's the ATN performance, and we have a minority of FTLD patients are correctly classified as SNAP. The majority are classified either as normal or, but if we simply replace p-tau and instead incorporate a marker that is sensitive to FTLD, light chain, and this was actually introduced in one of the morning talks about how it's also sensitive to ALS. We see that it's sensitive to FTLD, tau, and TDP. Now we can see that the detection of FTLD as SNAP is much improved. The large majority are classified as either SNAP or Alzheimer's disease and SNAP. I'll point out also that when you compare the proportions of AD patients who are classified by ATN using tau and ATN using NFL, these proportions stay largely the same. So you're not losing information. There's not a downside when you're classifying AD. I do not pretend, though, that NFL works really well in all SNAP cases, and that's because SNAP is this umbrella term for everything that is not Alzheimer's disease. And so that, of course, includes a lot of pathologies. This is not exhaustive. Each of these have their subpathologies. There's just a lot of pathological heterogeneity under the umbrella term of SNAP. And so I think finding biomarkers that are sensitive to each of these is actually going to be useful instead of just trying to find one that meets the needs of all. The second source I'm going to talk about is the fact that biofluids are influenced by patient factors other than the main pathology that they're thought to measure. So I'm going to focus largely on p-tau because, again, T status is necessary under the 18 criteria to meet a definition of Alzheimer's disease. So I'll focus on p-tau. What I see is that p-tau is not equally sensitive to all forms of Alzheimer's. Here's a study in, again, autopsy cases. These are all AD patients. But what we can see are that CSF p-tau levels are significantly lower in those patients with a non-amnestic or atypical phenotype compared to those with an amnestic phenotype. And this is despite the fact that we see that both groups have widespread tau indicated by the Barak stage of 3. In this other study, I look at how p-tau levels vary in patients with pure AD compared to those with co-occurring alpha-synuclein. And when we look at the relationship of CSF p-tau to tau pathological burden and alpha-synuclein pathological burden, we can see this positive relationship with tau, which is what we expect. That's what we think we're measuring. That's great. But when we control for the levels of tau, we see this negative relationship with increasing alpha-synuclein burden. So this means that patients with AD with co-occurring Lewy body disease are more likely to have lower levels of p-tau, more likely to be T negative. And that's important because Lewy body disease is one of the most commonly co-occurring diseases with Alzheimer's. And I guess my point is with this is that it's really important for us to know the blind spots of our markers. So this is a study looking at how CSF p-tau levels vary across a couple of different studies. This is a meta-analysis in African Americans compared to white Americans, and they show that p-tau levels are lower in African Americans compared to white Americans. We also see that t-tau levels are also lower in African Americans compared to white Americans, and they're also affected by other patient factors like sex. And both of these are affected by age. And the study by Barnes that I showed you earlier also shows that African Americans may have levels of alpha-synuclein copathology in AD. So I don't mean to indicate that all these factors aren't non-overlapping, and I'm curious to know the relationships between each of these, but I think it's important that we know where the susceptibilities are. And this is also not to say that p-tau and t-tau aren't useful markers in AD. There's many, many studies showing that when they're combined with A-beta levels, they really increase the specificity of the AD diagnosis. It's simply that I think when we correctly identify which populations they might be low in consistently, they're going to perform even better. I'll point out that CSF A-beta is not influenced by these patient factors. It's neither influenced by race, based on this meta-analysis, and my previous studies looking at age, phenotype, and alpha-synuclein copathology and sex show that A-beta is invariant to all these factors, which is another way of saying that it's equally sensitive to these populations. So I think these are all lessons I'm taking with me as as the field moves towards plasma-based biomarkers. There's a lot of studies that are excited about specifically p-tau 217 and 181. They do a nice job showing discrimination of AD from non-AD, but a lot of these studies are validating plasma by comparing it to CSF or PET. And I think it's important that we continue to validate using autopsy because we don't want to just recapitulate the errors of other biomarkers. And some of my preliminary work shows that plasma biomarkers are susceptible to other factors that we haven't seen in CSF. So I think it's always nice to come back to the pathology, always validate it with the pathology. And this is just, I'm just going to highlight this nice community-based study that I think does a nice job looking at the biomarkers in plasma and how they stratify patients with high AD neuropathologic change, intermediate, low, and not. And they also look at different subpopulations to see if the predictive value is maintained across different populations. So thanks for your attention. I am very grateful to everyone who helps me with this work and has contributed both within the FTDC and beyond. Thank you for your attention. Yeah, that's one of the things I subtly was thinking about was a lot of these things are kind of jumbled together, and I don't know which factors are, they're all correlated. I don't know which are the driving factors when we look at phenotype. Non-amnestic phenotypes are more common in non-white populations, even though they're still less common than the amnestic. And I talked about like alpha-synuclein, Lisa Barnes showing that that's more common. And so it will be curious to me, I would love to have the power and the sample and the diversity to really disentangle which of the factors are the driving and for the lower p-tau levels. But yeah, even when I look at patients with high levels of AD neuropathologic change, with high levels of tau accumulation and high levels of A beta, we still see this negative interaction with alpha-synuclein, and I kind of wonder what's going on there. It's one of the factors I'm more interested in exploring. Let you get out of it.
Okay, so next is Dr. Emily Largent, who's Assistant Professor of Medical Ethics and Health Policy at the Perelman School of Medicine and a Senior Fellow in the Leonard Davis Institute of Health Economics.
You do anything?
He doesn't.
Oh, it's magic.
Yeah.
Okay. Great. Well, Dr. Bennett, when he was here, he said that he was worried about being the odd man out, and so added in some pictures of scans and other things. I have to say, as the ethicist, I'm always used to being the odd man out. Instead of having any graphs or images, I rely on stock art photos of older adults. And the other thing that always makes ethics somewhat unique in this space is that after a day like today, you have heard about so many different scientific advances and the amazing work that's happening at Penn, thinking about how this is going to affect patients' lives. And ethicists have a little bit of a reputation for being a killjoy, and I am here to play that role and just say that we have amazing scientific advances, but society is really lagging behind where all of you are in thinking about these issues. And so we have ethical, legal, policy challenges that need to be addressed in parallel with all of the advances that you're making so that we can have a maximal impact on how patients are doing once these things make it into the clinic. So I'm glad I followed Dr. Cousins because her talk about biomarkers really set this up. So we have a sense now with the various frameworks that we can identify Alzheimer's disease pathology well before the onset of cognitive symptoms. And so we can tell patients about their risk using various biomarkers. So this creates a really interesting moment for individuals, because when we think about, in the popular imagination, what it means to have Alzheimer's disease, people think about dementia, and they think about that as a disease of autonomy. They lose the ability to make decisions for themselves, to exercise decision-making capacity, and do things like that. But what we're doing now is we're actually going in and we're telling people well before the onset of cognitive impairment that they are at risk for cognitive decline, and can understand how that starts to affect their thinking. So we have individuals who don't have a lack of decision-making capacity, which is often treated as synonymous with autonomy, but we've given them information that actually already starts to buffet their sense of autonomy. So I'm going to talk a little bit about results that we have on what it's like to learn that you are at risk for Alzheimer's disease due to genes or biomarkers, and then say a little bit about the policy changes I think we need to make sure that we're ready when this hits the clinics. So one of the first things we know is that once you give people information about genes and biomarkers, they will tell you consistently that this is unlike learning any other medical information they've ever learned about themselves. And there are 4 key areas in which they say this is true. So first off, this has unique implications for their identity. One of my favorite quotes is that somebody said, well, this isn't a colonoscopy, it's my brain. So that role in identity is foundational. They immediately see this as having interpersonal implications. They start to think about how friends and family might treat them differently once they know they have this information. People might see you as less of a person. They talk a lot about the uncertainty of outcome. So today we've talked a lot about uncertainty and how we can't explain everything with genes and biomarkers, and people are very well aware of that, that this is something that might or might not happen, and it can be years into the future. The final thing they talk about is that there's really nothing you can do about this information right now. So we did a lot of the interviews that I'll be talking about today before the approval of Aduhelm. I think there's still not a lot you can do now that we have Aduhelm, but people speak to that. And for many people, that's actually a reason not to find out their risk, because if they can't do something medically, they're not really interested in finding out about their risk factors. Now, while it's not medically actionable, people do come back to us and tell us that, in fact, it's empowering to have this information because they can make non-medical changes in their life. So it's not a drug or a pill or some other sort of prevention strategy, but they will tell us they change their health behaviors. We have people who start running again. We have people who will go on and on about how much they hate salmon, but now they're eating it twice a week because they've heard that the omega-3s are really good for their brains. They like playing Sudoku. They're learning additional languages. They're doing things to try to maintain their brain health. People also start to see their future differently once they have this information. So people who find out that they're at increased risk for dementia will often tell you they're using that information to plan ahead. So we often see people thinking about long-term care insurance. They're thinking about trade-offs between work and retirement. Do I stay longer for the cognitive benefits and the social engagement that I get that might help me? Do I try to save money for future care needs that I might have? Or do I retire now when I'm still able to check things off that proverbial bucket list? So people really do find that to be an area of tension. We also see updating wills, writing advance directives, and talking with family members about end-of-life wishes. We see people who downsize their houses and also move closer to, say, an adult daughter who will be their most likely caretaker. This information can also change how people perceive their own memory. So for people who find out they're at less risk than they thought they were, this is comforting. They reinterpret memory changes as just normal aging. But for people who find out they're at increased risk, they often talk with us about having this experience that now they're taking these senior moments and wondering, is this how it starts? And we have a lot of very haunting quotes where people are starting to question and worry when they forget things that they might not have worried about before. People talk a lot once they find out they're at increased risk about the possibility of stigma and discrimination. This comes up in 4 key areas. One is in their interpersonal relationships. So maybe their kids take away their car keys sooner or won't let them babysit a grandchild. They're worried about people losing confidence in their abilities at work and perhaps being overlooked for new opportunities for advancement or new projects they would like to be part of. Certainly in insurance purchasing, and also in housing, say purchasing into a continuing care community, they worry that if people know they're at risk, that will be a strike against them. Unfortunately, those concerns are likely quite merited. So this is a study that was led by my colleague, Dr. Shana Stites, in which she presented vignettes to people in the course of a survey. And what she found is that just having a positive biomarker, not having any sort of cognitive impairment, just the fact that somebody has a positive biomarker will actually create a greater reaction of stigma to that individual. Now, we know that strategies to manage stigma and discrimination are unfortunately quite limited. The one that people predominantly rely on is that they engage in what I call selective sharing. So they go through a process of trying to decide who they're going to share this information with so that they can protect themselves. And what we see here is consistent with what's true of other non-visible health information, where people consider the recipient and whether or not they'd like to share with them. So you're likely to share with your spouse, but not with your estranged brother. You think about what their possible reaction is. Are they going to offer you support, or are they going to be upset by it? And you think about your own efficacy to deliver the message in a way and at a time that's comfortable for you. For people who are at reduced dementia risk, this is like no news. You know, one of us even questioned when we asked who they were sharing with. They're like, well, you don't go around telling people you're not diabetic, so why would I go around telling people that I don't have biomarkers? But people who had increased dementia risk, some of them actually made it. They drew parallels to coming out of the closet and other sorts of like deeply revelatory experiences where you share information with others. As for legal protections, this is one of those areas where I'm going to be the killjoy. We have the Genetic Information Non-Discrimination Act that offers some protection here. I would say it's very inadequate. It varies somewhat also by states. California has much stronger genetic protections than do other states. We have nothing for biomarkers. There's no BINA. So if somebody has a biomarker that puts them at risk for Alzheimer's disease, we right now don't have protections for people when we send them out into the world with that information. Monitoring is an area that I think we need to watch in terms of potential conflict within families. So some adults, but a small percentage who are at increased risk, say they would like other people to watch them for incipient changes in cognition. And some family members, not necessarily the same family, tell us that they start looking closer at the older adult. And this is a place where we hear concerns about intrusions on privacy, restrictions to ability to do things. And what's going to start happening too is we have these digital biomarkers. So we now have good evidence that you can start to see before the onset of cognitive impairment that people's banking patterns change, their driving patterns change. And as you think about this kind of health information in the hands of non-health actors, Geico is not a healthcare provider, not covered by HIPAA. We have to navigate how we're going to handle that. We also know that 1 in 5 individuals who have biomarkers tell us they'd be interested in medical aid in dying if they were to develop cognitive impairment or if they were to feel like they were burdening their family members. Most of the people that we've spoken with, despite very high educational levels, are not aware that they would not currently qualify for aid in dying. So this is, I think, we'll have a growing constituency of people who are interested in but don't have access to. So this is an area to watch. So autonomy ultimately is not just about whether or not you can make choices, whether you have capacity. It really is about the context in which you are able to make those choices. And I think there are some ways that we can try to protect, promote the autonomy of older adults who are at increased risk for developing dementia caused by Alzheimer's disease or any of the dementias we've talked about today. And some of these things are going to be developing new social narratives to reduce instances of stigma. We need to strengthen anti-discrimination laws, particularly with biomarkers in mind. We need to think about the ethical and legal implications of digital biomarkers. This is an area where really not anything has been done. We need to think about what it means to live and die with dementia, and so who should be eligible for aid in dying, and maybe we want to expand to people with neurodegenerative diseases, although that obviously comes with challenges. There will need to be new paradigms for getting your affairs in order. We have well-known limitations in advanced directives for handling the medical needs of people who might have dementia for, say, a decade. So we need to think about how we can take this bridging time where people are not cognitively impaired and help them through the trajectory of cognitive decline, not just at that very end piece where an advanced directive would normally come into place. And ultimately, we need to increase resources for healthcare because we know that preclinical Alzheimer's disease is estimated to affect something like 45 million older adults. And so this is a huge number of people who might benefit from some of the precision medicine that Dr. Bennett described, but that will be with huge demands on the healthcare system and huge resource needs. I'd like to give the last word to one of our participants. Gina Collada wrote this article asking whether or not people should get an Alzheimer's disease test, and unbeknownst to us, somebody who had been in some of the research here at Penn wrote in and talked about their experience. This individual had learned that they had a negative amyloid PET scan, but he says, if the results had been different, I would have accepted them with the knowledge that I could plan for my ultimate demise in a way that would work for me and be helpful to my loved ones. When we disclose, we really do have to disclose with sensitivity, but trust that people can use this information in ways that are meaningful to them. Thank you so much. You're so good, Dave.
Well, I'm kind of curious about that. Result they lose their key 70, they assume it's Alzheimer's. 30, I just lost my key.
Unless you're in our line of work, in which case. Yeah.
But so I mean, is your sense or both of them changed?
Yeah. So this is, with the qualitative research, we can't make any claims about causation, and I think there's a tendency to assume that once we give negative risk information to people, that's why we see more changes in that group, more exercise, more fish, more Sudoku. But I sometimes wonder if we've actually had a negative effect and suppressed advanced care planning and other things in people who now feel falsely reassured. I mean, people can tell us, they know intellectually that this means that they are less likely to get dementia caused by Alzheimer's disease. And most of them can tell us, well, I know there are many causes of dementia. And yet they're like, but my future is wide open. I'm not cleaning out my attic anymore. You know, this is great. And so I do think that's an area for future study, is to figure out, are we suppressing change or are we driving change? Paul? No, either. I saw two hands. Yeah.
Like, is there any?
Yeah, so I think there are some laws that might come into play and help out here, but people who would assume, for example, that HIPAA would cover this are going to be woefully mistaken, you know, again, that these things are not covered. And so it's really a new area where we have to think about what we do, and not only how we protect the information, but what are the ethical and legal obligations of, say, a bank once they realize that you are overdrawing your account regularly or you're missing payments, to either notify you or others. And you know, that's not an institution that we normally think of as being well suited to disclose medical information. So lots of questions about the relationships there and how we need to protect them, and also what obligations are within them.
In our experience with familial forms of FTD, the genetic disclosure result is very parallel to what you're describing with biomarkers, and talk about the genome rules and how inadequate they are. Yeah. Some of our family members who are negative, pre-symptomatic, who have the result, have a negative outcome where they have feelings of guilt and other consequences. I'm curious if you've experienced that in the AD space.
Yeah, so once we've given people an APOE result, we go back and have asked them, you know, have you told other people about this in your family? Have you encouraged them to undergo testing, and what did they end up doing? And a lot of family members received the information. Many actually declined to get tested. A few people, we had some E4 homozygotes. They told their siblings, you know, I think you probably have this, you should also get tested. And the siblings ended up getting tested and then declining disclosure because they felt so scared about their results. And then one of the things when people find out they are not a carrier, they tend to be so relieved, and they often talk about it in terms of the consequences for their family. Like, I can go home and tell my son I didn't give him this. And so, yeah, we see it on all sides, you know, people who do want to know, people who don't want to know, and people who are so glad that they can tell people that they don't have to worry. Great. Well, thank you so much.
Okay, so we're a little behind schedule, but I think we have time for a brief discussion if all agree. We're near the end of the day. So we have Dr. Emily Largent could join us, and Dr. David Woke, Director of the Penn Memory Center and Penn ADRC, Dr. Virginia Lee, the Director of the CNDR, and Dr. Lauren Elman, who heads the the ALS Center. So for those who are remote, feel free to write in questions. I think this can be very informal, and we heard a lot today. I think one theme that came up was the visualization of disease processes, and we heard from David Bennett how there's a large amount of cognitive decline and aging that's unexplained, and different ways to how to detect that. And this morning we heard from Leonard Petruccelli about ways of visualizing DPR for C9, and that might be a biomarker that could show target engagement in a clinical trial. So I guess our first question for the panel would be, and I think, thinking about how we planned this and how different the world is in perspective of COVID, but also we have an FDA-approved medicine for Alzheimer's disease, which I think many of us did not see coming, and it is clouded with different controversies. So do you view clinical trial results as an informative to disease mechanisms? Can we look at those as a way to learn about disease biology, or are there aspects of trial design that are flawed that may be hard to interpret?
On? Is this thing on? Yeah, I mean, that's a very, thorny issue. I mean, I certainly think well-designed studies that, target particular biology and that have outcomes that, influence disease trajectory, certainly can tell us about factors involved in the progression of the disease. I think the challenges are forgetting, the controversy around Biogen and how well designed. Well, the study was perhaps reasonably designed, but the behavior in terms of stopping the study early is a sort of a different issue. But I think that, what also comes up today, and particularly when we're trying to treat Alzheimer's disease, and particularly when that treatment isn't necessarily going to be something that absolutely halts the progression of disease, but that's going to be a more mild effect, we're left with the fact that even in patients with Alzheimer's disease, there's lots and lots of heterogeneity pathologically, perhaps in resilience, perhaps in a variety of other factors that creates a tremendous amount of noise in these clinical trials. And so, you have a high type 2 potential in clinical trials that you're not necessarily going to see an effect unless the study's done really well. There's lots and lots of participants. You follow people long enough to see those effects, and again, that you have a robust enough effect that within that noise that you're still going to see an outcome that is measurable. And so I think that's really the challenge in our field. On the other hand, I think all the tools we talked about today potentially allow us to better stratify or organize our trials in such a way that maybe we can increase our power to kind of get at those results and feel a little more confident in them.
Hi, I could comment on that a little bit. I come from the ALS world where sometimes, unfortunately, doing clinical trials feels a little bit like Groundhog Day, because we keep doing them and doing them and doing them and doing them, and nothing seems to work, and it's incredibly frustrating. Lately, there's been, a real interest in focusing on targeted therapies, which is much more satisfying because we sort of know what we're aiming at. Previously, and still to this day, it's a little bit of throw spaghetti at the wall and see what sticks. So I think, to be fair, that's very unsatisfying in terms of really making any comment as to disease mechanism, which is something that ILS scientists can sit around a table and argue about ad nauseum, and it's really not interesting at all. I'll be happy to tell you that. We've had some recent trials where they have been so-called successes, and there are sort of two very different stories here. One of those drugs, did you want me to talk about this?
Sure, yeah.
One of those drugs, Radicava, was actually FDA approved in 2017, and that was approved on the basis of a trial of a subset of people with ALS with a very specific phenotype, but it was given a broad approval for all people with ALS. And then the company did an extremely aggressive advertising campaign, which made it seem like this was the next coming, that this was going to save everyone, and people really wanted to take this drug. The interesting thing is, and I won't go into too much detail because it gets a little bit boring, but the question of whether something that's statistically significant is clinically significant becomes very, very relevant in this population, and in every population. In this particular drug, there is a lot of energy and effort required to take it because it's an intravenous drug that requires port placement, et cetera, et cetera. And if you take a careful look at the data, you will see that actually it's not a very clinically important benefit that patients get at all. But that being said, when the drug first came out, which again was in 2017, due to this very aggressive marketing campaign and a little bit of massaging of the data in how it was described, I will say it was not incorrect how it was described, but for lay people, it was very misleading. A lot of people wanted to take it. But in my experience from having cared for these people, sort of one by one, they kind of dropped off taking the drug because they found that the return on investment in terms of their time and effort was just not worth it. They were definitely not seeing any clinical benefit, which they never should have expected, but again, based on what they were promised, it wasn't happening. And they were actually working pretty hard to take this drug. So there was actually really a negative there. So I think that's a really important thing to weigh when you look at data because statistical significance does not mean clinical significance. Recently, there was a phase 2 trial, which we were involved in, of a compound called AMX0035, which has fairly robust results, not anything earth-shattering, but better than anything we've had to date. This drug is oral. It has very few side effects, a little bit of GI, very safe, no toxicities, no monitoring even necessary. And hooray, the FDA declined to approve it. So that's fabulous. And now they demanded a phase 3 trial, which we understand, but the company this particular company has done everything right. They continued to collect data from the open label extension of this trial, so they were able to add more data. And just about 10 days ago, the FDA allowed a resubmission. So we're waiting to see if this drug will get approved. And in this population of people, this oral drug that actually extends life longer than any other drug that has been approved for ALS is really something that the community wants to be able to take. And there are some other statistical niceties about looking at this data, which shows that earlier exposure to this drug actually improves outcome and things that really don't look questionable. So I think that I think I'm sort of echoing what Dave just said, is that you have to take a look at the specifics of the data. You can't just rely on statistical significance and other things about the drug to see if it's really going to benefit your community.
Right, but even for Alzheimer's disease, I mean, I think that for any neurodegenerative disease, and the biomarker that really stick that we have, A-beta and tau, which is basically the pathology, and so you can even explain why you don't see, other like synuclein or TDP in blood or in CSF, and in part because of the fact that there's not enough pathology to really kind of allow them to leak into the CSF, if you will. But A-beta and tau, because there's so much going on that they're just not degraded, and so you can actually measure them. So I think that it's a very unique situation. That's what I'm saying, is that maybe we would be a long time before we have new biomarkers that really truly perform and detect ALS or FTD and so on. I'm not very optimistic, but I think we need to try.
So in my field, we're still using clinical outcomes, a functional rating scale, which is a pretty gross way of measuring things, but it's the best that we have. So we all just throw our hands up and say, well, it's the best that we have.
No, that's great. So, I mean, I guess again, kind of getting back to this question of can we learn about mechanism? It's in humans, it's not mouse models, right? So it's hard to control for as many things as you can there. And I actually think even with biomarkers with amyloid and tau, which we can actually measure very well, we can see the pathology that a pathologist can see on some level. Even when we move those biomarkers, we still have to keep in mind that we're potentially kind of uncoupling the relationship of those biomarkers to the underlying biology with regard to what they mean. So, for example, I'm actually somewhat optimistic about some of these amyloid-based drugs that really clear amyloid because they really move tau in multiple different kinds of measures, so plasma, CSF, and to some extent tau PET. But I do worry that if clinically we don't see signal in those trials, then we might be having some sort of effect on the biomarker or way of measuring that biomarker that isn't necessarily reflected in disease progression. And so I think there's always a kind of caution in the things that we measure in vivo and what they mean, in terms of how they link to, the disease mechanism, and ultimately the proofs in some sort of clinical outcome. Although even there, you still don't know mechanistically for sure whether it was because it hit this target or it had some other sort of effect that you didn't expect it to have.
I have a related question for Virginia about mechanism. We talked briefly before, and I thought it was interesting about TDP-43 and how it has so many important roles for the cell. What do you think the best mechanism would be to try to treat a TDP-43 proteinopathy? And I guess the follow-up would be, because we see it with age in the context of AD, are there any model data that suggests that there's age-related factors that alter the propagation of TDP-43?
I don't know. Yes, I really don't know. TDP-43 just and you have to come up around, or identify some of the pathways further down, and maybe you can. It'll be a while before we can treat TDP-43 proteinopathy. This is what I just said.
Can I ask, do you have any opinion about Stackman 2 as an upstream in the upstream pathway?
Well, in the animal, it looks pretty reasonable, but I don't know. I mean, you guys are doing the clinical trial, so.
Well, not yet.
Okay.
I mean, I think it might be. You can make an ASO.
Right, but the ASO has been around, and then I think that, yeah.
Maybe, right? Maybe.
So, Emily, it was wonderful that you joined us to have the ethical perspective, and I can imagine that Aduhelm really changes a lot of the landscape of what you're working on. Have you encountered this in the context of clinical trials, the ability to remove amyloid from a living patient, and how that would affect decision-making and biomarker disclosure, and some of the effects, social effects from that?
So we definitely have seen that there are people who would only be interested in being tested if there was something they could do about it. So in a few cases, we had people who learned that they had a gene or a biomarker in the context of screening for a trial. Then they found out that they were ineligible for reasons outside of the trial. So there was an individual, for example, who had actually been a transplant recipient, and that transplant made them ineligible to be in the trial. And I still wonder why they couldn't figure that out sooner, but they had already disclosed, and he talked about how he felt like it was other people who weren't as strong as him, because he had such dark days after finding out the risk information and not being able to do anything, that he didn't think other people could have handled it the way he handled it. So, I mean, it's good, right? He didn't meet clinical levels of depression or anxiety, but, clearly having something you can do is going to make some people much more open to learning information that can be quite upsetting to them. And I'm also very interested just to see how things will interact, right? Because if you get, I would hope that with aducanumab prescribing that people will both be getting an amyloid PET scan and an ApoE result. I'm not necessarily required by the FDA label to do that, but those would be good clinical practice guidelines. And I'll be interested to see how people understand that information together. So we definitely have people who, once they find out that they are not a homozygote for ApoE, are like not even interested in hearing what the amyloid PET scan result was, because they're so reassured and so happy. And so we're going to have to think about how to communicate sort of multiple aspects of risk to people.
Could I ask a general question, actually, to Dave and Lauren, and in particular, Kurt? And he's looking at his phone. I'm actually making him think and put his have on. So you know a lot about, you have a lot of connection with people in the pharmaceutical industry. So other than, now A beta and tau are not the whole story, and it's kind of getting old. But what else did they do? And so this audience may be interested in terms of. Yes.
Of a lot of active investigation. I mean, so if you look clinically right now, at least for AD, and I guess for some of the other tauopathies, it's still tau immunotherapy is still active, but of course there's already been failures there in PSP, for example, and so that's got people worried. And now, Biogen actually withdrew from AD, so I mean, there's some concerns about tau in that sense. And so, they're trying to go other places now, and I think like TREM2, they're looking very closely at that. Other glial inflammation markers, but I can't say right now that there's a better, brilliant idea that I'm aware of that they're working on.
Okay. Yes.
Yeah, I have a brilliant idea. No. Yeah, I mean, I was just at the clinical trials of Alzheimer's disease, getting a little bit of the buzz around things. I mean, obviously amyloid still is a big target, right? And, I think until the current sort of swath of trials like donanemab, lecanemab, that have shown some promising data, until they fail, if they fail, I think that it will continue to be an area that people will invest in. And then if they do fail, then there'll still be questions of earlier treatment. Tau's also, I think we're at early days with tau. I mean, to get to where we are with amyloid at this point, it's taken over a decade or longer, and the first active immunization trials were 15, 16 years ago. So I think tau is just at the beginning through a variety of mechanisms, a lot immune based. There's many more sort of genetic based, ASO based, therapeutics. Lots, I think, of earlier phase stuff on microglial, inflammatory pathways, especially given the genetics of AD. And then I think actually there was starting to be some buzz that links a little bit to maybe what David Bennett was talking about around pathways that affect cell senescence and aging to influence, the biggest risk factor of these diseases, which is, aging cells, aging brain. And so I think that's, also potential therapeutic strategy that there may be some druggable targets there as well.
Well, they're the ones with the money, so. In the ALS world, there's a lot of focus on genetic ALS. So the order of operations has been SOD1 first, C9 ORF72 is second, and ataxin-2 is third in terms of first to the finish line. We just had terribly disappointing results on the ASO against SOD1. Early phase data was extremely promising. The phase 3 trial, however, failed to reach its primary endpoint. And the hand.
Even with the one that they SOD1, because
yes,
yeah, okay.
Yeah, in SOD1 patients.
In
so
SOD1 patients,
yeah.
okay.
So yeah, so the good news, bad news, depending on how you look at it, and I'm still not sure which side of the fence I'm on, is that they had excellent target engagement of the ASO, so it did reduce SOD1 protein expression in the CSF. So the drug did exactly what they hoped it would do. So you can't say that the drug didn't work, so you can't go back and fiddle with the drug and just say that was the problem. It seems that the problem probably is that we just didn't get these people early enough because they were already symptomatic. These people are being followed in open label extension to see if they do diverge from the controls, which may happen over the longer term. But I think that the money is going to be in treating the pre-symptomatics, and that study is already underway, treating pre-symptomatic SOD1 patients with the ASO against SOD1. The C9 program follows the same pathway as the SOD1 program, so the phase 1/2A of that program is in the open label extension now. The phase 3 is planned for early 2022. The Ataxin 2 trial, and you should know, by the way, that was discovered here in Alice Chen Plotkin's lab with, I forget it, Aaron Gittler was, that was his work. Yeah. Oh, I'm sorry, Aaron Gittler's lab, sorry. That was his work on yeast, originally identifying that as a risk factor for ALS. And we here at Penn have just discovered that it is heritable. It is a heritable trait. So that's groundbreaking stuff. And we're doing the first in human trial on that here. And if that's okay, that'll go to phase 3 as well. So that's the story there. In ALS, we're looking for anything that you can make an ASO to, because we have already done our dance with the anti-inflammatories, and it's really a fantastic failure. A C5b inhibitor trial was just stopped premature, or not stopped, it was stopped for futility, I should say, so that's not premature. But nonetheless, another one just completed data. Are you going down that road as well, C5b inhibitor?
No, I was just going to say, futility analyses based on that. It's hard to say whether it's premature.
Well, I'm quite sure this one was futile. Yeah, sorry. So lots and lots of anti-inflammatory stuff has been tried due to imaging that shows microglial activation. The problem in ALS, which you may also have in your diseases, is that many of us think that there are inflammatory stages that may occur in ALS, and it may be worth treating people with appropriate anti-inflammatories when they're in the inflammatory stage, but not otherwise. And we don't have the biomarkers to determine when they're in the inflammatory stage. So we're kind of just like, hitting a pin with a mallet, and that isn't working. So that's sort of where we are with that.
Lauren, in the ALS pre-symptomatic, we have a similar situation with FTD where families now have a very different decision-making process about getting genetic testing. What are the patterns you've seen, and how trials may or may not be affecting genetic testing?
That's a great question. So we are now, I will just say it, super aggressive about offering and encouraging testing in every single newly diagnosed patient, whether they have a family history or not. And the discussion centers around the fact that there are these trials either available now or coming online for pre-symptomatics. And we're really not honestly focusing on the fact that someone identified with a genetic form of ALS may be helped by a trial, because the truth is nobody really believes that anymore, sadly, but that it may be important information therapeutically for family members. And we're getting, I think, upwards of 80% of people are now having genetic testing. And it isn't just for those genes that I mentioned for which we have therapy, it's for a whole panel. And that has been a real sea change in terms of not only what we're offering, but also what is happening. And a lot of the credit for that goes to our genetic counselor, specifically Lainey Dratch, who's my cousin. I'm very proud to say. I say it all the time because I'm so proud, because she works with the patients and gets it all covered by insurance and everything, which is makes a huge difference, and discloses results, and is really a fantastic resource for our patient population.
This is a question for Dave about, it seems like amyloid modifying therapies are going to be in the clinical research space for a while now. How does that affect research? It seems like we still have a lot of natural history data that we don't understand that we need to collect. And is this something that enhances our research, or something that's an obstacle, or both?
I think Emily can also maybe talk a little bit about this as well. But I think there are probably challenges and opportunities around approval of Aduhelm. Right now, I think as an impediment to research, unless there's a sort of sea change in, people's view of the medicine and the sort of enthusiasm around it, I suspect it won't have a super large effect for many of our observational studies, just because I think, right now people are a bit allergic to prescribing or taking the drug. That may be different actually, like, for example, we're doing the longitudinal early onset Alzheimer's disease study. I think if you're 55 years old with Alzheimer's disease, your sort of risk tolerance for a drug that may or may not work might be a bit different. I think it will be a challenge if donanemab and lecanemab get approved, which are sort of the next two drugs going in. Well, at least lecanemab's officially gone in for accelerated approval, and donanemab is probably behind that. On the other hand, I do think one important point is that if the drugs show efficacy, so let's say donanemab does show a clinical effect consistent with its phase 2 study, I think the fact that a drug might affect clinical research isn't a reason not to have it approved. And so I think I would like to be in the position where we have a drug that we're very confident about having a benefit if it impacts our clinical trials. But I think I think it will be a challenge. I think it'll be a challenge for clinical trials, but certainly a challenge for observational studies and sort of the natural history of the disease. On the other hand, it may also bring in more people to, both from a funding standpoint, from an industry standpoint, continuing from a federal standpoint, but also from a patient and, for example, we follow a lot of preclinical people, that there'll be a lot more interest in participating in research in the field because, of the potential therapeutics. I don't know if you want to comment more about that.
Missed a comment from Eddie Lee about other targets for neurodegenerative disease. He mentions that proteostasis is being targeted, things like PROTACs, integrated stress response pathways, and that PROTACs induce targeted degradation of target proteins. ISRIB inhibits the interrogated stress response, which may target multiple neurodegenerative diseases. Several screens are ongoing for molecules that affect chaperones, disaggregases. So that's interesting, of these other upstream mechanisms that could affect multiple neurodegenerative diseases.
So that the major problem with that approach is that, you're turning everything off. And is it a good idea? I mean, I don't know. So it's really hard if you want to try to, downregulate, like you said, the inflammatory pathway. And often that type of approach could create more problem rather than benefit.
I guess one question I had, that was loud, maybe for Virginia, Dave, or Eddie, if he's listening in, is to, and we've sort of talked about this in the past, this issue of polypathology, there are multiple pathologies. To what extent do we feel that those, or does the biology support the idea that those are independent processes that co-occur in the context of aging, whether they have some sort of more common upstream mechanism or whether they sort of interact with each other? Because that may influence where you want to target in terms of thinking about these more sort of further upstream events, or at least we have some data in our brain bank that you don't tend in AD to see that much of the TDP and alpha-synuclein until a little further along in the disease, which then would be an argument for earlier targeting of the disease. But again, if it's from a sort of common pathway before that, then you might want to treat that common pathway before that. So I don't know if you all have thoughts on that.
Yeah, I mean, I'm leaning more toward not a common pathway, but, certain things could lead to others. Like, for example, particularly when you have Alzheimer's, and it's a bad neighborhood already. So things are sort of not going well, things are degenerating in the brain. And then you kind of get, other pathology. The cells is injured. So they would mount other response. And one of the response may be to develop a Lewy body, particularly over 10, 20 years, right? So those things we don't understand right now, but that's kind of my bias right now, rather than the fact that they're all from one source and then,
Yeah, I would agree. I think that several from CNDR shown, I think Mako might have some data on increasing Brock stages and late TDP43 pathology. Sanaz from my lab has looked at ghost tangle markers and finds that that correlates with TDP-43, so suggestive of the same kind of more advanced or more severe cases happen to have another pathology, but what that mechanism is, it's upstream or not, is unclear.
I guess the other sort of model that suggests that too is, the autosomal dominant AD familial forms, and then actually early onset AD that much to, I think, surprise of a number of us who think of those as pretty pure. There's a lot of alpha-synuclein in particular in those cases, actually pretty high rates, depending on the study you look at.
Does anyone in the audience have any questions for the panel? Here we are. Oh, Eddie has a question. Most people think the entire system that maintains proteostasis declines with aging, but I do absolutely hear Virginia's concern about non-specificity of ramping up protein turnover in general. So these are complicated issues for a Friday at 4:30. Everybody, this has been really an exciting day. I'm just so happy to see everyone. There's so many of you I haven't seen in months, and this has been a wonderful session. I think we can conclude. I can announce poster winners. So for the basic science, Song Her's posters, slow motor neurons resist pathological TDP-43 and mediate motor recovery in the RNLS8 model of amyotrophic lateral sclerosis. I don't know if they're here today or remote, but congratulations on the poster, first prize. And then for clinical posters, Pavan Vaswani did serial olfactory testing for the diagnosis of prodromal Parkinson's disease and the Parkinson's-associated risk syndrome. So similarly, Pavan, congratulations here or in cyberspace. So really would like to thank all the speakers, our panelists, Dr. John Trojanowski, Virginia Lee, and all of you for attending. And to Kathy and her team, did enormous amount of work behind the scenes to set up everything today, including Ebony Fenderson and Nicole Calvecchia, who changed names between now and the pen. I think she got married and had a kid somewhere in the interval since the last time I've seen her. So thank you to everyone and have a wonderful weekend.
Thank you, David. Dave, you've done a terrific job, so we thank you too.